Hello. Okay.
Hello.
Yeah.
A very good morning to one and all present here. I welcome each and every one of you who have joined us here for the second International Conference on Clinical and Applied Microbiology on the theme Sustainable Future Through Microbial Innovations, held on the 14th and 15th of December. The conference is organized by BioLEAGUES with alongside our research partner, United Innovators, our academic partner, Singhad College of Pharmacy, and our media partner, the Leaders Magazine USA. On that note, I would like to welcome the keynote speakers for today. Professor Dr. Mohammad Nazir Mohammad Desa, Head of the Department of Biomedical Sciences, Faculty of Medicine and Health Sciences, University Putra, Malaysia, from Malaysia. The next keynote speaker, Associate Professor Dr. Priya Madhavan, Head of Non-Communicable Diseases and Public Health Research Group, School of Medicine, Faculty of Health and Medical Sciences, Taylor's University, Malaysia. Ms. Dilara Monpitia, Medical Microbiology Associate Practitioner, University Hospitals, Lando, Public Health Wales, United Kingdom. Professor Dr. Sandeepta Vergula, Head of the Department of Microbiology, Director CFRD, Osmania University, India. I would like to welcome the session chairs for today. Dr. Debashis Banerjee, Assistant Professor, Biotechnology, Atmaya University, India. Dr. Swati Kumari, Assistant Professor, Microbiology Department, Sri Ramachandra Medical College and Research, India. Dr. Sinchan Das, Founder, Benevolentia Foundation, India. Dr. Amit Chauhan, Coordinator for the Department of Forensic Science, Christ University, India. I would like to thank each and every one of you for your participation and for your encouragement. And we truly hope that this session today is going to be a fruitful one for all the participants. On that note, I would like to invite the first keynote speaker, Professor Dr. Mohammed Nasir Mohammed Desa, for the keynote lecture. Sir, I would like to initially thank you for accepting our invitation. And participants, if you have any queries or any questions from our to ask our keynote speaker, I request you to put it in the chat box, or you could raise your hand to ask a question. Sir, I welcome you for your lectures.
Thank you. Let me share. I'm sharing my slides now.
Yes, sir, we can see the slides.
Okay. So can you see my slides?
Yes, sir.
Slide. Okay,
Yes,
thank you very much. Okay, thank you for the invitations to deliver a keynote lecture on this platform. I'm going to talk on the, my topic is microbial genotyping in the genomic era. So I'm going to talk on the history, the developments over a century, now with the genomics and digitalization technologies that we have, that have shaped the direction of microbial genotyping in the genomic era. So, and then I'm going to share a little bit on my experience working on Streptococcus as a model of organisms, particularly Streptococcus pneumonia and some other Streptococcus species as well, if you got times, to see where the organism have served as a good model and the feasibility of the genotyping against these dynamic organisms. Okay, what human civilization have achieved back in the last 100 years, more than 100 years back, till now has been tremendous. And we can see, starting with the simple experiment and tools, now with the highly sophisticated machines, coupled with the computer algorithms through various analytic software that are able to analyze huge biological information that are translated into digital codings to comprehend microbial behavior. So something that was not probably imaginary in the past, but now they are real. And this started with the discovery of DNA and the biological descriptions of the double helix structure of DNA somewhere in 1950s that have revolutionized, the research direction as well as the applications. And along the way, there has been a lot of innovation in instrumentation and computerization. So my research started in 2000 when I was a postgraduate student, primarily in the field of molecular epidemiology in Staphylococcus and Streptococcus species until now. So this is the data that drive the innovations. So with the decline of the theory on spontaneous generations, Those who study microbiology would find this in the textbook. The microorganism has been recognized to exist and has been the causing agent in disease. There has been causing agent of diseases in animals, humans, and plants that lead to from low to severe disease manifestations. Since then, the classification of microbes took place that led to the field of microbial taxonomy. Then the specific nature of infectious diseases are assigned to the specific group of organisms, the species, then followed by the specific guidelines for control, prevention, and treatments that are tailored to suit the behavior and infection or transmission pattern of that particular species. So now we are in the 21st century. So what are the leading organisms? So based on these publications in 2022, there are 5 most leading species in causing infectious diseases, which are Staphylococcus aureus, E. coli, Streptococcus pneumoniae, Klebsiella pneumonia, Pseudomonas aeruginosa. I have been working on Streptococcus pneumonia in particular within the scope of molecular epidemiology. And I will share my work in the next slides later on. And we can see many cases are taking place in these 2 leading regions, Sub-Saharan Africa and South Asia. So the question is, if you are talking about a species in general, is a group of bacteria belonging to a single species equally pathogenic? That is the question. So, well, it's not likely because there are strain-to-strain variations, and the more invasive or highly associated with drug resistance, or probably both, probably it just involves a subpopulations of a species that eventually manage to spread along the transmission lineage, carrying certain beneficial genetic traits that make them more successful in initiating colonization and infections. And the persistence of the lineage are also being facilitated by many external factors, such as and there are multifactorials, it can be a demographic backgrounds of the host, the environment, the socioeconomic of the populations. So therefore, if we look at the graph here, what would be the scenario in these 2 regions, in these 2 leading regions, to explain the high incidence of infection? So there is questions. So this is the field of disease epidemiology. And of course, with the molecular technologies, with the advancement of molecular technologies that further enhance the investigation by providing evidence at the genetic levels. So now we are able to trace the evolutionary lineage of the strains, and with that we are able to answer a lot of important questions related to the epidemiology. So the genetic traceability, okay, is determined by microbial typings, or to be more specific, the microbial genotypings. So the outputs of genotyping is the discrimination or differentiation between strains, so that we know whether a group of strains are highly or closely or maybe distantly related, or probably not related at all. So if they are highly related, most likely they are descendant of the same origins. So with this information, we will be able to answer the critical epidemiological queries, such as what are the relationship between the different isolates. So what are the source of the different strains of an outbreak, whether the second episode of disease is due to a previously isolated strains or not, or perhaps some other strains, or whether an infection is caused by more than one strains. Okay, and then it will also allow us to monitor the prevalence or occurrence of certain related strains within a settings, and it will allow us to investigate if a cluster of infection are unrelated or part of an outbreak. So these are actually a very important question that we need to answer, so that later on we can come up with the management, we can come up with the programs to manage and to come up with the proper prevention programs. Okay, so what are the approaches of microbial typings? We know actually, as a microbiologist, we are familiar with the phenotypic methods. We all know about it, which are actually time-consuming, not discriminatory, time-consuming. At the end, it's not confirmatory. So you can imagine the time wasted doing so many, probably require so many tests to be done, and eventually there's no guarantee that you will be able to actually discriminate the strains. So molecular approaches that make use of the DNA is more discriminatory and accurate. Okay, some method visualize the output in the form of DNA band fingerprints generated by PCR or recession enzymes that can be compared through some computational analysis within a specific setting or a collection or within a collection of samples. So there are still associated weaknesses, especially in terms of the reproducibility and data portability. And then we have a DNA sequence method, DNA sequence-based approach such as MLST and WGS, whole genome sequencing, which are more reproducible and accurate, and most importantly, they are portable. Okay, because the data are translated into digital codings where this code can be stored in the database and everybody can get access and retrieve and do comparison within our to be compared with our local isolates. So it will allow comparative worldwide analysis through web-based database. So this sequence-based methods has been widely utilized because of these benefits. So it has been, since then, it has been widely utilized. Sometimes they are coupled with the traditional PCR approach, depending on the specific questions of interest. Okay, and then we take a look at the genomic texture of bacteria. So we are talking about bacterial genomes, not the eukaryotic genomes, which are more complicated. But bacterial genomes, prokaryotic genomes, particularly bacterial genomes, they are quite simple. But it's not simple as it's not simply saying simple. You mean that is less complicated to study, but actually, there are a lot more complex. Actually, there are some textures, some organization in the genome actually that we can manipulate for many purposes. For example, here we have a coding sequence, we have non-coding sequence. So some methods make use of the coding sequence, such as MLST. Some make use of the non-coding sequence that are being repeated, such as the repetitive elements, the palindromic sequences. So these are repetitive sequence that can serve as a target, as a marker to be amplified by PCR. Eventually, we can generate the DNA fingerprint as well, such as RAPD, ERIX, box, and some make use of the presence of recession enzymes, such as PFG and RFLP. So, but actually none of these methods has all the desired attributes. So it's very important that the choice of typing methods, as long as we understand the scopes and limitations, the strength, the scope and limitations, as well as whether we are capable of executing those methods, because different laboratory may have a different resources. Okay, and we may not have expertise for all things, so that will depend. And then the most importantly is that we have to set a question of interest within the scope of our capability. So although we can claim that certain method has better outputs, but sometimes we are not able to do it due to the financial constraints, lack of expertise, and so on. So we can decide which ones are actually more appropriate to suit our questions of interest here. Okay, so a little bit on the history. With the discovery of DNA, and then lead to the developments of DNA sequencing. So the technique to sequence DNA has been developed somewhere long time, a few decades back in 1960s. So, but of course, the process is very lengthy, and probably costly. But now there has been a lot of improvements where sequencing can be done faster with high specificity. And now the whole genome, the whole bacterial genomes actually can be sequenced rapidly, especially with the introduction of NGS, new generation sequencer. Even now we have another. I can't remember the brands actually. There are new versions of platform that actually it's more rapid. Okay, and of course the cost might still be a concern. Plus we need the software, software and operators, bioinformaticians to handle them. And I think there is also a lack of standardized workflow on what to do on the biological data. There's so many software, but which one is more decent? So there's no standard to say that you have to stick to one software or not. So everybody is like the bioinformatician, they are publishing a lot of papers on their own software. So which one is better? Actually, we have to evaluate, we have to do assessments by testing on our local isolates, and then we can compare with the other studies to see the feasibility of that particular software. So the genotyping approach that are targeting limited genes such as MLST are still in use, because I'm also struggling to get familiar with the recent technology in whole genome sequencing, because I was actually trained as a microbiologist during that time, not as a bioinformatician. So it's a bit difficult, to explore new things while you are actually at the same time you are handling a lot of research that require your expertise, particularly to maintain, to isolate, to preserve, to maintain the biological creatures, these microorganisms. So it costs a lot of time. So sometimes we don't have time to explore. Perhaps the new generations who, from the beginning, they started with the computers, with the software. Perhaps they would be. We need them, to help us. Okay, to at the end to achieve the outputs. Especially the integrations of the biologicals, the biological platforms with the digital platforms. So it has a huge potential. So, but I can see now the MLST, the traditional MLST is still in use, and the outcome are still relevant within the scope of the analysis. Okay, this is actually a publication by a recent publication that did a review, a very comprehensive overview of the available typing methods. You can see here, there are actually a long list, which started with the phenotypic approach. And probably the phenotypic approach, although they are important to answer certain things, but eventually we have to couple with the more recent technologies, especially those that are working on the DNA sequences, because they are more reproducible, they are more sensitive. And the best model is actually to integrate both. So it will give a better picture of the overall relationships by having the phenotypic evidence as well as the genotypic evidence. So I have experience on some of these, such as RAPD, PCR, RAT-PCR, PFG, MLST, MLVA. Yeah? So if you look back in the history, PCR was invented in 1980s. So that somehow, with the ability of PCR, the thermocycler that we use actually are carrying out the PCR. It has revolutionized the various PCR-based typings. But I admit that based on my own experience, that to maintain the reproducibility of the PCR-based typing is quite challenging. And at about the same time, PFG was invented and claimed as the gold standard due to its reproducibility and discriminatory power. So it used precision enzyme to cut various sites of the whole genome to generate quite a comprehensive and a stable genome patterns. But it is tedious. I got experience on PFG. It's very tedious and time-consuming, and works comparatively well within a specific setting, specific sample collections. So they are not portables unless if we can pull all samples from various geographical locations and subjected to under a single protocol in a single lab. So that is the challenge. So later comes the MLST in 1990s that involve sequencing approach. It amplifies around 5 to 8 housekeeping genes depending on the species. And then with the advancement of sequencing technology, such as NGS, so MLST begins to get a wide attention due to the data portability, where we can do sequence comparison worldwide by getting into the web-based database. And it's very good for a long evolutionary analysis because it works on the slowly evolving housekeeping genes, while PFGE might be best in a short-term outbreak. But definitely the combination of both actually will provide a more insightful story of the microbial transmission patterns. And then the MLST involved PCR and sequencing of a number of housekeeping genes, okay, to determine the allelic variations that are translated into allelic numbers and the sequence type, okay? And this serves as the genetic lineage ID, something like an ID or nomenclature of these strains. So it is not tedious as of PFG, especially now with the availability of molecular kits. We have DNA extraction kits, PCR master mix, as well as the availability of the service provider. There are a lot of sequencing service providers that are actually offering a cheap cost to do sequencing. So of course, at a certain level, it will cost you more, but it is made feasible with the decreasing sequencing costs using the latest platforms such as NGS. So previously it was expensive, but the price is decreasing. But of course, it's still expensive as compared to some other methods. But it's worth considering the quality of the data. Okay, and then this is an overview of the methods that are getting the popularity. PFG is still there because it's previously considered as a gold standard. But now there is not much talking about PFGE, because we are more excited with the potential of whole genome sequencing, the WGS, where we can mine and analyze all genes of interest that we want using available bioinformatic pipelines or custom analysis to suit our specific needs. And of course, the genes included in the WGS will automatically cover all the genes that we work in the traditional MLST. But of course, in WGS, we got more genes. So we can add more housekeeping genes that are available in the WGS. So that's how now we have core genome MLST, CG MLST, or whole genome MLST, which works on more number of housekeeping genes. It can be 100, 200 genes, 300 genes. So, but the question is how many genes to be considered as sufficient for the MLST analysis. So previously in the traditional MLST, we work around 5 to 8 housekeeping genes. But would the numbers simply provide the same resolution? Probably if you work within 5 to 8 against 100, against 200, but eventually we will get the equal resolution, then probably we don't have to waste money to do WGS. We can stick to the traditional MLST. But this is just a question that we have to, we need a lot of studies to compare whether to establish the universal, the feasibility of the traditional MLST over the WGS. Of course, theoretically, if you're talking about WGS, it's a huge information. There are a lot more genes. Theoretically, it should be better in terms of the resolutions, but it has to be tested and approved based on the actual experiments. Okay, and then of course, eventually, what we want to tell from this analysis, normally, we want to classify.
virtual background virtual background.
Sorry, we want to classify the strains on how they are related to one another. So this will help us to see the dynamic of the transmission lineage or patterns. So these are some of the common terms that we use to describe the level of relatedness. So starting with the sequence types, okay, the MLST will give the sequence types, and then we can do a downstream analysis to find out the colonal complex, the colonal groups, eventually the colonal complex, and eventually the clades at the larger volume of populations. Okay, and eventually, we can answer so many things, particularly we can apply MLST to answer questions in the field of molecular epidemiology and public health, in the field of phylogenetics, taxonomy, and speciation, as well as questions in the field of population structure and dynamics. Okay, so then the WGS. Okay, technically it looks complicated, it looks complex, but it is actually an automated process. So actually the software will do the works, as well as the downstream analysis also is simply automated. So, but we need to interpret, the output, and this is the job of a microbiologist, probably not the job of bioinformatician. Because microbiologists, they understand better the microbial behavior, because we are dealing with the live organisms. While bioinformaticians, they are good probably in constructing the computer commands, but actually to answer questions that are coming from microbiologists, the question come from us, so we need them to construct the commands towards answering our queries, our questions. But there are also, we can still ask whether the software are accurate or not. There are so many free software, whether they're very decent one, we don't know. So we need to accumulate the data and compare whether they are really feasible, whether they're very accurate or not. Because from my experience, there is a pipeline where we can use to mine to determine the serotype, but there has been errors being reported. So we still have to double check by going to the gold standard method using the antigen reactions to confirm. So there is still questionables. And then, of course, it's highly computational, so they require skills. And of course, the genome assembly can be fraught with difficulty, which might introduce errors as well. And of course, we don't realize it, and probably we will consider the errors as something that are actually belonging to the strain-specific variation, why it is not. Okay, so this is the issue. And in order to reduce the error, to ensure the reliability, we need a good reference genomes. We need reference genomes. Okay, so we need to have a reference genome in the gene bank so that everybody can go and access and utilize this reference genome to be aligned against the world type isolates. And there is also still lack of standardized workflow in the NGS, in the WGS. So in order for the WGS-based method to become a new standard in strain typings, so there must be a full confidence in the accuracy and robustness in the data generated across different sequencing platforms and laboratories. So there also, another thing is that also depends on the nature of the specific species. You know, I have been working on Streptococcus pneumonia, which is actually, it has open genomes, meaning that it got a lot of homologous recombination, a lot of mutational events, which that will actually make things complicated a little bit if you want to study the evolutionary lineage of this organism, because too much recombination, too much mutation might lead to some wrong assumptions if you want to trace the evolutionary line, because it does not reflect the evolutionary lineage due to this extensive recombination. So, but some organisms, they have a closed genome. For example, like Microbiology, the genome are very well conserved. So we don't have to worry about, you know, filtering out all this recombination, all the genomic part that contain the extensive recombinations. So, but as far as Streptococcus pneumonia is concerned, we have to filter out, meaning that we have to do a lot of assessments, bioinformatic assessments to remove all these mutational parts, and working only on the highly conserved, which are the housekeeping genes. So that's why earlier in the earlier slide, I show the approach. One of the approach are actually to either going into the core genome or whole genomes. Okay, core genome just focus on the highly conserved housekeeping genes. Of course, there will be hundreds, but if you're working with the whole genome, they will include all these, you know, accessory genes which may be altered or changed due to the mutation or recombinous homologous recombinations. So that requires skill as well. Okay, so then I would like to share, this is the scenario related to the genotypings, from phenotypics to PCR-based to sequencing, and now we have whole genomes. It sounds good, but of course it requires a lot of assessments, you know, before it can be very well established as a gold standard to replace all the traditional methods. So I have been using the genotyping working on Streptococcus pneumonia. And so Streptococcus pneumonia is actually an old pathogen. So it has been well studied long time ago, more than 100 years back. And in fact, it served as a models to demonstrate the natural transformation. One of the good properties, one of the miracle properties of this organism is that it is naturally transformable, where it can simply pick up the DNA fragment environment and incorporate into the genomes. And that's how they lead to the homologous recombination. So that's how we are dealing with a lot of, you know, mutated part of the genome that lead to the emergence of drug resistance, enhanced virulence capacity, and so on. And somehow they find a way to escape from our prevention program, the escape from the vaccination program, and so on. So the genome is the genetic plasticity of Streptococcus pneumonia is very dynamic, that put up a lot of challenges in tracing the evolutionary lineage based on the whole genome sequencing. Medically, it is one of the important organisms. It is one of the leading cause of pneumonia, especially in the high-risk group, children and elderly. That's how it got the name Streptococcus pneumonia, because it's very famous in causing invasive disease, especially pneumonia, and also meningitis, the brain infection, and so on, otitis media. So the genomics and genetic of Streptococcus pneumonia, the first complete genome was published in 2010. The strain, this is an old strain that actually the earlier scientists in 1960s worked on. the Fredericks, the Avery et al. And so on. Most of the reference genome we have now are actually originating from the earlier works during that time. So you can imagine the time spans. Although now there are a lot more genomes of Streptococcus of the more recent actually are being deposited into the gene bank, but these old genome are still there to serve as a reference. Okay, so there are, as of 2018, there are about 27 complete pneumococcal genomes. And of course, there are a lot more draft genomes in the data bank. So these are the list of a complete genome of pneumococci, which can serve as a pool of available reference genomes for the downstream analysis of the WGS. And this is the this is an old story actually, but still relevant. We are still referring to this. The earlier the establishment of the PMEN, pneumococcal molecular epidemiology network, that was established in 1997, where at this time probably they don't have so many complete genomes yet, so they are relying on PFGE, MLST, and some of the PCR-based typing such as bulk PCR, as well as some of the phenotypic approach like antimicrobial susceptibility patterns, and so on. So they come up with actually we need to come up with a certain system for assigning the ID for the genetic lineage so that we can trace over the times by giving something like an ID. We call it nomenclature, so that it will be easy for us to trace the existence of that particular genetic lineage that are reflected by the ID nomenclature over times. So that's what we do. We're still doing it until now. So this is the criteria. Okay, there are criteria, and eventually this is the naming systems. Okay, the country that was first identified, the first identified serotype and the country, and then the for example, if it was first identified in Spain, so they put Spain there, and then the serotype, and then they put number 1, okay, because it was the first to be identified, and then the if there is another emergence of the same lineage but carrying a different serotype, they just put a different number, another number to differentiate that this is belonging to the same lineage, but now they're carrying a different serotype because of the capsular switch events. Because the same origin, the same lineage of populations, previously they're carrying, for example, 19F, serotype 19F, but along the way, the same lineage, they switched to 19A, for example. This has been reported because of the vaccination pressure. So they want to escape from the vaccination pressure, they switch to another serotype, which are no longer covered by the particular vaccines. But of course, now we have a new regimen of vaccine that are targeting a wider coverage of serotypes. So starting with the PCV7, pneumococcal conjugate vaccines, now we have PCV10, PCV13s, and now this is going to be PCV20, it's still under clinical trials. Okay, so this is how we name it, so that it can help us to trace, to help with the traceability. So this is some of the successful clones established during the 1980s and 1990s. Okay, there are currently more than 40 clones described by the PMEN network. The first 16 are published by McGee et al. In 2001. And then clone 17 to 36 has been confirmed by the network. But the problem is they need the type strain to be submitted. So somehow it just stopped there until clone 36. And then we got no news what happened to clone 36. Clone 37s. Probably there's some legal issue about sending off their isolate to other country. That would be some possibilities. So, but with this nomenclature, with this system of nomenclature, then we can it will facilitate the traceability. For example, we can see the prevalence of certain clones. For example, Spain 33F. ST sequence type 81, which are disappearing, but there are certain clones we are still persisting until now. Okay, so I will share my work where there are certain clones which are still persisting. And this clone was first identified back in 1990s, 1980s, but now we are still getting that particular clones. But some clones are disappearing probably because of the effect of the vaccination. Okay, PMEN networks. This is the old platform, but now we have a new platform. We call it Global Pneumococcal Sequencing Project, where all these clones are being transferred into the new website platforms, but they are still the same clones. Okay, so and they remain available, for to be referred and to be cited. And in fact, we are still referring to this when we found the kind of clone in our collection. We still refer whether these clones are still related to the old clones. So they are still being cited, although these are this actually went back in 2000s, which is now we are in the year 2023. It's more than 20 years now, but we're still referring it. Okay, this is an example of my earlier work in 2000s. At this time, we were still using PFGE, yeah? But we can see the patterns of the clonal dissemination. This is actually isolate collected in a single hospitals. We can see there are two groups of clonal groups carrying 19F, serotype 19F, and they are penicillin resistant. So whether they belong to the internationally circulating clone, we don't know because we don't do MLST. So then to answer that, so in the next few years, we use PFGE and also MLST, involving an archive collection of pneumococcal isolates of serotype 19F, okay, isolated from a number of hospitals throughout the different part of Malaysia. So it's very interesting to see that the clone, the ST236, which was first described back in 1990s, which are related to Taiwan's settings. So Taiwan 90F number 14, that was the nomenclature given back in 1990s. And we still find these clones disseminating in Malaysia. In fact, actually, they constitute about more than 50% of the collection. We have PFGE, but PFGE look a bit diverse, probably because of the time span of isolation over 3 years, and it's coming from different parts. So as I said earlier, PFGE is better, is good for short-term outbreak, but over time, of course, as I said, pneumococci is open genome. They keep mutating. That will somehow alter the genomic, the genome parts that will alter the genome of the organisms. But MLST is working on a slowly evolving housekeeping genes. MLST is still able to cluster all these, probably the group of organisms coming from a single source, limited source, and evolve throughout time, but they're still related at one point. So you can see MLST is still able to cluster them until at a single point here. And they're all belonging to the same sequence types, ST236, which are actually related to the Taiwan 90F14 clones. So this is good things to find out that this particular clone has been persisting over how many decades, so many decades. And we are very interested to know what would be the nature, the features of the genome somehow that allow them to survive over a long period of times. Okay, and then recently the pili, actually pili is not common in gram-positive, but it was reported to be present in a population of the pneumococci. So perhaps pili would be the added advantage. Okay, if that particular strain is carrying pili, they would have a extra beneficial. They would have a certain advantage to survive because pili will help them to facilitate the adherence. So we are interested to see whether the occurrence of pili or pilated isolates actually, whether it's a random or probably because of the clonal factors, okay, that are related to the globally circulating clones. Okay, so this is a review where we found that actually all these majority of the pilated isolates are belonging to the globally circulating clones. So that would explain that probably pili would add the survival advantage by facilitating the cell adherence. That would explain why some of these globally circulating clones has been so successful, in the persistence. Okay, and then we conduct a study to see whether you will be getting pilated isolates. We do actually. This is a study that involved isolate from a major tertiary hospital in the east coast of Malaysia. So this is the results. You can see the MLST, they clustered them in a single clade 1, okay, together with a few other STs such as ST320 to form a colon complex groups. So interestingly, all isolate that carry complete pili gene regimens are they clustered together. Okay, and these are actually a group of clones that are related to the globally circulating clones, and they are carrying pili. And the same observation also when we do another study at another hospital in the West Coast. So we have the same patterns. Okay, or the we don't have ST previously in the hospital in the East Coast. We have 236. Okay, previously in 2014, we still find 236. Now in this study is taking place in 2018, 2019, and published in 2020. We still find the ST236 and they are carrying pili. And then in another study on another hospital in the West Coast, we don't have a 236, but we do have 320, which are belonging to the same clonal groups. They are related. 236 to 320, they are related. They are part of the clonal complex. But unfortunately, we don't have so many samples from the hospital. Previously, in the East Coast, we have more than 100 samples, but somehow in this hospital is only about 40 plus, not so many. Probably because the West Coast are a lot more developed, a lot of hospital here. So the patients are not concentrated on single hospital. They go to different hospitals. So that probably explain why we are getting low number of infection, because they are being, perhaps there are other infection case in the other hospital as well. So that is the limitation. So of course, to take advantage of the WGS, we from the isolate collection, we select some representative to do the WGS. This is still an ongoing work. We can see basically the genome size, the GC content do match with the pneumococcal characteristics, pneumococcal genome characteristics. This genome size is about 2 megabase pair. GC content is about 40%, which is consistent with the previously reported features of the pneumococcal genomes. We haven't done much analysis yet, but preliminary we did a co-genome phylogenetic analysis based on the whole genomes to get some idea, to get some clues what we'll be dealing with. So we can see here, it's quite interesting to see that so those that are in bold are actually our isolates. Of course, previously there has been some pneumococcal genome from other studies deposited for representing Malaysian isolates. So we can see, obviously, there are 3 major clades, 1, 2, and 3. And it's very interesting to see that for clade 1, you can see these local isolates are related to the South Korean and Taiwan's. This is consisting the old clones, which are 236 and 320. These are the clonal complex, which, as I said, they has been persisting for many decades. Until now, they're still around. And there's another group which are more related to the European isolates. We have Denmark here, Norway, Italy. So this is very interesting. And then another group related to Lebanon, the Middle East. So somehow the Malaysian isolate are very diverse, coming from different continents. That's true probably because we can see actually we got so many foreign students coming to study in Malaysia, and there's so many travelers coming in, as a tourist. So we have a very dynamic pneumococcal populations. It's a mix of local strain and international strains. Perhaps there will be new emergence. We do find a new sequence type, but not so many. But we do find quite a number of unique new sequence types, which have yet to be reported elsewhere. Okay, when we blast in the MLST database, the curator will have to, when they find out that this is not there yet in the database, so they assign a new ST. But not so many, but we do find some, probably this is a local isolates. And then of course, the we're talking about pneumococci now with the current efforts by the governments, to the government has incorporated the pneumococcal vaccines, the PCV10. So it's very important to do the surveillance to see the patterns of serotypes occurrence, and then of course the serotype we have to do MLST genotyping, and then compare with the serotype to monitor the potential incidence of serotype switch, because the current vaccines are targeting only limited number of serotype, those that are commonly circulating. So it's not comprehensive enough. So we have seen the emergence of, for example, with the introduction of PCV7s, which include serotype 19F, because we know that serotype 19F is very common, but it does not include serotype 19A yet at the times. So when the PCV7 have been widely used, there has been emergence of 19A. And when we do the genotyping, we find out that 19A and the 19F are belonging to the same lineage. So these are the same descendant of origins. Somehow previously they are carrying serotype 19F, but now they are expressing 19A, and they are actually belonging to the same groups. So that would indicate that there has been serotype switch, it's the same lineage that switched from 19F to 19A to escape the PCV7s. So now we have PCV10, which include 19A. So Malaysia is using PCV10. So we have to keep doing the surveillance to see perhaps there will be new emergence of, because we believe that PCV10 would be effective based on the epidemiological data that would cover largely the commonly, that would cover the most common circulating serotype in Malaysia. But of course, that will probably be temporary. Eventually, we will be expecting the emergence of serotype not covered by PCV10s. And probably at the time, we will have to switch for PCV13s or PCV15 or PCV20 in the futures. So this is very important work to keep continuing doing the surveillance by performing serotyping and genotypings. Okay. And then a little bit on the my work on the group B streptococcus, because just to see the potential of genotyping to help us to trace the origins. This is Streptococcus group B, which a lot more easy to handle as compared to the Streptococcus pneumoniae. We are very fastidious. So there has been an outbreak in Singapore, and they found that it was caused by group B, which is not very. Group B is very common among pregnant women, not very common in the healthy adults. But this infection is involving healthy adults, and it was associated with the eating fresh, they are eating raw fish meats. That's just probably the culture among the Chinese. They like to eat. In Singapore, they like to eat the fresh raw fish meats. And actually, a group B streptococcus is also very common in fish, in the freshwater fish. It's a big industry in Malaysia. So this organism has been causing death that has resulted in economic loss. So there has been a group of people working on group B strep, but among fish, while I has been working on human isolate all this while. Suddenly I encountered these incidents. So our groups try to see whether we probably, it happened in Singapore, which is our neighbor. Perhaps we should have GBS that originate from fish that has jumped into humans. We don't know because we haven't done any study yet. So we conduct a study to see to do the assessment to see the potential presence of GBS. And with the genotyping, we know that actually the one that caused the GBS, the one that caused infection in humans, are belonging to a lineage ST283, which are not common in human, but they have been very common in fish. And in fact, in fish GBS, mostly they are all belonging to ST283. And a few others, but mostly they're all 283. So something like it's all the same origin that are causing problem in fish, not in humans. In human, they're a lot more diverse. We got so many kind of ST. And then with the outbreak from Singapore, it was caused by the GBS of ST283. So now we are conducting a study in Malaysia to see. We are collecting so many samples from a number of hospitals. At the same time, we are also doing genotyping among fish GBS. And we found that all the fish GBS are exhibiting ST283. And among the humans GBS, they're very diverse. But we do find two strains that have ST283. So this is very interesting, meaning that somehow we believe that this is probably originating from fish settings and it have jumped into humans, but it have not caused a severe infection, but that will just simply put us on alert that it happened in Singapore. If we don't do something, it would happen here in Malaysia as well. So this is a paper that first reported the occurrence of SC283 in human. There are two of them. Perhaps if we keep continuing with the surveillance, we'll get more. We don't know. So this is something that we are doing. To collect to as part of the monitoring purposes. Okay, so what can we do? So, well, what can we do? If you go to the public and talk about these things, probably they're not interested. It's too technical. But at least actually, what can we do is just very simple things, because we need to minimize, to reduce the risk of transmission, to stop the spread of the clonal groups. We know a certain clonal group, they're associated with the drug resistance and high invasive capacity and so on. So we have to slow the transmission, or perhaps we want to eliminate them out. So, well, simple thing that we can do, just take care of our hygienes, wash your hands, cover your sneeze, take a proper medication, take vaccines and so on. That's it. Very simple things. So that's all from my lecture. Hopefully it will, useful for the audience. Perhaps somebody might be doing the same thing, that might want to initiate some collaborations or whatever in the future. Thank you very much.
Thank you, sir, for an interesting lecture. Participants, the floor is open for discussion. If you have any queries, you can drop it in the chat box, or you can unmute yourself to get your doubts clarified. So we will give a couple of minutes to see if anybody has any queries.
No questions. If you have anything, you can email me. You can get from the secretariat. You can email me anytime. So.
Thank you, sir. Thank you very much for accepting our invitation, sir, and also for graciously letting the participants know that they can reach you. Sir, actually, we do have a question, sir. The question is, for identification of strains, do we need to go for whole genome sequencing?
Well, practically, we can identify, depends on what kind of organisms. Certain organisms can be easily identified by biochemical tests. Okay. So, of course, you can identify by WGS, but WGS, you have to spend a lot of money. At the end, we just want to identify. So that will depend on what you want. If you want to identify, then there are options actually that will save a lot of money. Yeah? But later on, you want to identify, at the end, you want to study the genome, then you can do WGS. But actually, it's still expensive actually. So before we spend money on expensive method, why don't we, just to ensure that we are actually, when we do a WGS, we are sure that this is the organism, okay? Rather than simply without knowing what kind of organism, we go straight to the WGS and eventually it happened to be not the organism of interest. So you are wasting a lot of money. So at least you do some conventional way to get some clue that this is the right organism that you're supposed to work with before you go to the WGS. Definitely the WGS can help to identify, but it costs you a lot. Okay, so we have to think in terms of the practicality and cost effectiveness. Thank you.
Thank you, sir. I hope that query was clarified. Yeah. She's put that, thank you. Thank you, sir, for clarifying it.
Thank you.
Thank you once again, sir.
Thank you.
Sir. For the next keynote lecture, it would be of Associate Professor Dr. Priya Madhavan. from the School of Medicine, Faculty of Health and Medical Sciences, Taylor's University, Malaysia. Unfortunately, she was unable to be present at the moment because of travel, but she has given us her recorded video. So we will watch the video.
कैसे हाँ पहले थोड़ा फर्क है ना तो परसों से समय तो बारी बारी से
Hi participants, I think there's been some error with the audio. Please give us a couple of minutes to have it sorted out.
parasites. These microbes are collective of indigenous environment on environment. Good morning, everyone. The title of my presentation today is the effect of socio-demographic, lifestyle behavior, dietary habits, and living environment on the gut microbial diversity of indigenous Orang Ulu in Sarawak, Malaysia. I'm Associate Professor Dr. Priya Madhavan, and I'm from the School of Medicine at Taylor's University. So the human gastrointestinal tract hosts bacteria, fungi, viruses, and parasites. These microbes are collectively referred to as gut microbiota. These organisms have a symbiotic interaction with the host, in which the host offers nutrient-rich environment, and in exchange, the microbiota provides benefits. So the factors affecting gut microbes are diet, lifestyle, environment, host-related factors, and also drugs. Therefore, the aim of this study is to study the differences in microbial diversity and its associated lifestyle factors among Kayan and Kenya, who are also known as Orang Ulu. These are the indigenous communities from Sarawak, which is a state in Borneo of Malaysia. So we look at the method. So this is the workflow of the study. The first part of work is the field work. The ethics approval was obtained by the National Medical Research Registry and Taylor's University, Malaysia. After getting the ethics approval, the demographic data and stool samples were collected from an indigenous community living in long houses in Sungai Asap, Belaga Town, Bintulu, Sarawak. The stool samples were collected using Omnigene gut tubes. The second part of work is the lab work. So the second part begins with the extraction of DNA from the stool samples using QIAGEN stool DNA extraction kit, followed by purity analysis of the extracted DNA using nanodrop. After checking the purity of the extracted DNA, it was then stored at -80 degrees Celsius till further DNA sequencing. The third part of the work is the bioinformatics work. So this was based on the analysis of DNA sequencing. The DNA samples were sent to Apical Scientific, which is a sequencing company, for 16S rRNA gene sequencing to get the raw reads. The raw reads obtained from the sequencing company were completely analyzed, including the statistical analysis part in our labs using softwares, namely Quantitative Insights into Microbial Ecology, known as QIAM2, and RStudio platforms. The final part was based on the statistical analysis, followed by report. So the target community were people known as the Orang Ulu. This is the indigenous community from Sungai Asap, Belaga, Sarawak. So this is a picture taken from the longhouses, and this is also the original picture captured during the sample collection for this project in 2019. So this is the demographic distribution of Kayan and Kenya ethnic groups in Sarawak, Malaysia. And if you look at the red boxes, so Kayan was the most number of people, and there were majority females, and also from the age group between 50 to 60 years old. So this was the household income. Majority of them had below 800 ringgit per month as a household income, and a majority of them were non-working. However, some of them were farmers and fishermen, and the rest were students or others. And this was their BMI, and their BMI was of normal distribution, majority of them, and the education level, majority of them were only educated till primary school. And this is the taxonomic. So this slide actually shows the relative abundance of the phylum and genus level. So for part A here is looking at the phylum level. So majority of it is from actinobacteriodata, bacterioid data, cyanobacteria, desulfobacteriodata, and so on. And in B, you can see the most abundant genus were from Eubacterium, Alloprevotella, Bacteroides, Escherichia, Shigella, and so on. So these are the most abundant that we got among these two groups of indigenous community here. So the next one is on alpha diversity. And in alpha diversity indices across dietary related factors. First of all, as per our analysis, the ethnicity, BMI, gender, occupation, household income, education did not reveal any significant association with alpha diversity. However, we do observe a significant association of dietary patterns with alpha diversity. So here in figure A and B, the number of meals, timings of meals, and bowls of rice exhibit significant association with alpha diversity. Diversity was significantly lower in those participants having more than 3 meals per day. For timing of meals in a day, the results reveal that participants who mention having meals in the morning, afternoon, and evening had significantly lower diversity and evenness of bacterial species. And bowls of rice were also found to be significantly associated with alpha diversity but have similar evenness. Participants who normally took less than 1 bowl of rice had significantly higher diversity than compared to those who took just 1 to 2 bowls of rice in their meals. The next one here shows livestock association or related to pets or livestock ownership against the alpha diversity. Factors including an appropriate number of years of animal ownership, cleaning of animals, place of animal defecation were significantly associated with alpha diversity as well. As per results shown in A, participants having animals for an approximately period of 2 years exhibited a lower diversity when compared to the rest of the groups of participants. However, we did not find any association with the evenness of species. For cleaning of animals, in B, alpha diversity was significantly lower in those who did not clean their animals as compared to those who cleaned their animals. Moreover, the place or animal defecation also showed significant differences in alpha diversity. People who mention that their animal feces indiscriminately have the highest alpha diversity among the rest of the groups. So in this diagram, here we can see that when we tested for health status on alpha diversity, we observed that alpha diversity was significantly higher in those who had received antibiotics during the last 3 months of sample collection. For the type of toilet facility they are using, as we can see in B over here, these participants who are using both types, pit latrine or pore flush latrine, have more diversity as compared to the others. So these are the notes or summary of what we just saw in the previous slides. So here in table 2, this is association again with alpha diversity with respect to the socio-demographic factors, lifestyle behavior, dietary habits, and living environment. So the table is a summary of all these values shown. So this table 2 shows a summary of all the values including significant and insignificant for alpha diversity indexes with respect to the socio-demographic factors, food and water consumption, personal hygiene, pets and livestock. So for the analysis of beta diversity, we have employed Bray-Curtis dissimilarity index and weighted unifrac distances to investigate the structure of gut microbiota, and the principal coordinates analysis PCOA plots of beta diversity were generated using Bray-Curtis and weighted unifrac distances. So as per the results, there were significant differences in the gut bacterial diversity when considering participants who consume supplements as compared to those who did not consume supplements, and participants with probiotics and without probiotics. Participants having a number of meals as well as timings, and participants having different toilet facility. However, for all these factors, the amount of variability explained was low. However, you can also see in all these diversity images, there were no remarkable separations or cluster formation that was observed between the study groups as shown by the PCOA analysis. So this table shows a summary for all the values, including significant and non-significant values for beta diversity indexes with respect to socio-demographic factors, lifestyle behaviors, dietary habits, and also living environment. And then we come to the analysis of composition of microbiomes. So we have employed analysis of the composition of microbiomes known as ENCOM to plot volcano plots to know which features were more differently abundant among the study groups. And from all the factors tested for ENCOM, only 6 factors show statistically significant features between groups. For the factor of household income, only one differentially present feature from the genera Privotella was identified between the groups. Mitsuokela maltacida was differentially abundant in those participants who consume antibiotics as compared to those who did not. For supplement intake, Olsenella was differentially abundant in that group that consumed supplements during the past 3 months. However, Bilophila and Ruminococcus species, which were differentially abundant among those participants who had constipation problems. In this slide, ENCOM results for bowel movement per day reveal uncultured Ruminococcus species that were differentially presented in those participants who mentioned no bowel movements daily. Moreover, the place for animal defecation also showed significant ENCOM results. Uncultured Clostridium species were differentially abundant in those participants whose animals were defecating indiscriminately, that means in a random manner. So the study reflects the bacterial data dominated microbiota in these subjects. ethnicity, gender, BMI, household income were not the determining factors for gut microbiota diversity and composition as well as observed in our population. Prevotella, Faecalibacterium, Succinio-vibrio were a dominant microbiota in the Orang Ulu subjects living in the longhouses. Prevotella in abundance indicates that Orang Ulu follows a vegetarian diet. Moreover, the larger proportion of Prevotella was most likely caused by the consumption of ulam. Ulam is also known as raw vegetables. It is eaten like a salad. Faecalibacterium and Succinovibrio abundance indicates a diet rich in plant-based. Depletion was reported in association with different inflammatory conditions. As per the analysis, animal ownership showed a significant influence on gut bacterial diversity. A significant association was also observed between approximate years of animal ownership and the gut bacterial diversity. An interesting observation is that these populations have almost no lactobacillus and bifidobacterium genera in the microbiota, even though they have this traditional culture to drink this fermented rice wine known as tua. Escherichia shigella were also identified among the top 10 dominant taxa in the Orang Ulu population. And this is a matter of serious concern because these are associated with bloody diarrhea. The dominant representation of Prevotella, Faecalibacterium and Succinovibrio in the Kayan and Kenya gut is very appealing. It represents a potential source of healthy gut microbiota in fecal microbiota transplantation-based therapeutic strategies, especially in Malaysia, as they are involved in the production of short-chain fatty acids, the main metabolites produced by these microbiota in the large intestines. So this is the first of its kind to study and analyze the microbiota of the indigenous community of Sarawak and understand the link between the environmental and lifestyle factors and their gut microbiota. Rural communities also provide an excellent model to understand the influence of both natural environment and communal living on the microbiome. Yet, we hardly know anything about their microbiome. So the rapid urbanization in Sarawak is pointing out that it is high time to document the relationship between the biodiversity and the gut microbiome. Otherwise, these unique microbiome profiles will be lost. This is also because of the rapid cultural and socio-economic transformations that are happening in this state of Malaysia. These are my references, and I would like to thank you for your time in listening to my presentation.
Hope that it was a fruitful lecture for all the participants. Our sincere thanks to Dr. Priya Madhavan for accepting the invitation to be the keynote lecture and for sharing the video despite her busy schedule and traveling. On that note, I would like to invite the next keynote speaker, Professor Dr. Sandeepta Bargula, Head of the Department of Microbiology, Director, CFRD, Osmania University, India. I invite you to deliver the keynote lecture.
Good morning, everyone. I hope I'm audible.
Yes, ma'am. Good morning, ma'am.
Good morning. Thank you for inviting me to deliver this lecture. I can share the slides from here, right?
Yes, ma'am. Yes, ma'am.
Okay. Yeah, so today I'm going to speak on haptoglobin and its a role in acute phase response and diagnostics of sepsis. So before we understand what sepsis is, it is important to know that sepsis is a big life-threatening medical condition characterized by overwhelming infection, and it is simply systemic inflammatory response, and it happens most of the time due to the normal flora available in the body, in the gut, on skin, nose, upper respiratory tract, and so on. And most of the time, this sepsis is because of the failure of the immune response of the patient, and also the vital organs, which causes 50 to 60% of mortality rate for this condition, and which is quite high compared to other diseases. And the problem with sepsis is that there are several inducers of inflammation which cause sepsis, including the toxins released by the pathogens, or even opportunistic pathogen like E. coli, Klebsiella, Staph aureus, and so on. And what they do is they cause release in the cytokines, which are inflammatory in nature, and several chemokines and lipid mediators. And what happens is these cytokines cause an uncontrolled immune response, causing inflammatory response throughout the body, leading to damage of vital organs. And this occurs, as I said, due to release of these chemokines and cytokines and lipid mediators into the bloodstream. And this leads to a cytokine storm in the immune system, which leads to a hyperinflammatory state, causing C-reactive protein, which is a marker of infection, and STREM. So many mediators are elevated in this condition. And in sepsis, the infection is usually targeted by using antibiotics or antivirals to treat the patient. But whereas the cytokine storm has to be controlled by the immune system of the patient. Otherwise, it may lead to mortality. And this happens on 40 to 60% of the time. So sepsis can occur suddenly, and most of the time what happens is it can be diagnosed in the diagnostic centers by one of the best markers of sepsis, which is serum procalcitonin, which is used widely now, and C-reactive protein, which is only a marker and inflammation, but not sepsis. And the diagnostics of sepsis, we have observed that when we screen for the survivors and non-survivors of sepsis, the serum samples did not show much changes in C-reactive protein in all these cases. C-reactive proteins are serum procalcitonin or serum lactate, the WBC count, all these survive or not survive. Hence, we were looking for more about how we could preventing severity of sepsis in the patient, and how the patient responds to infection and inflammation in terms of their own immune system. So for this, the main reason was because sepsis diagnostics itself takes some time. It takes half a day to 48 hours. And what happens is most of the time the antibiotics which are used are not going to really affect the treatment of the patient. And the major focus we had on this study was to devise early strategy to diagnose sepsis and the risk of severity of the patient. And also, our aim was to map the protein profiles of sepsis patient to see if there is any difference in sepsis, and if any biomarker which can be identified can be used further for study. So basically what we have done in the study is we have taken serum of sepsis patients who could survive and who could not recover from sepsis. And we have separated the serum samples to two-dimensional electrophoresis at a 3 to 10 pH gradient isoelectric focusing strip. And when we superimpose the images expressed, in survivors and non-mass spectrometric analysis, we could observe that among these, most of them are involved in acute phase response, which controls sepsis or immune response of the patient in infection. And also several proteins control the coagulation and complement pathway in the patient. And among these, we found haptoglobin as most interesting because it is a acute phase protein, which is elevated. Although the basal levels are much lower, around 14 mg per deciliter of blood, whereas in females, the basal levels are 35 mg per deciliter of blood. So we found that the expression factor for this, when we compared the ratio of this haptoglobin protein volume on the gels, we found that the non-survivor to survivor ratio is quite low, meaning that it is very less in non-survivors compared to survivors from very early stages. And we found this result to be quite significant. And the other genes, as other proteins, as you are seeing in the table, transthyretin, S100A9, ORM, alpha-1 antitrypsin, serum amyloid A, SAA, as you can see in the bottom, all of them have shown a differential expression in survivors and non-survivors. And these findings were also confirmed by real-time PCR analysis of the respective RNAs for these genes. And we found that mRNA expression at early stages showed decreased haptoglobin levels and increased levels of alarmins, which are negative acute phase response proteins, which negatively affect the immune response in non-survivors. And we've also observed that pro and anti-inflammatory cytokine levels were higher in non-survivors, indicating that there is a high rise in inflammatory response, but it cannot be controlled in such patients. So the conclusion of this part of our work was that haptoglobin, mRNA and protein levels decrease very early in the early stages of sepsis, particularly in non-survivors, whereas it is the reverse in survivor patients. This means that its levels are inversely related when compared to all it for further analysis. So we observed that haptoglobin could be an attractive biomarker, especially since it has been studied previously in antimicrobial activity, and it is also a known neonatal marker for early onset neonatal sepsis. It has a role in antioxidant activities in the mammalian system, and also it is important in autoimmune response and tumor angiogenesis. Basically, what does is binds to free hemoglobin in blood, and this free iron bacteria or pathogen invading pathogens to multiply, therefore preventing any bacteremia and the multiplication of bacteria in blood. And next, it was difficult to get more and more patient samples, and we wanted to study how this finding of haptoglobin being elevated in survivors of sepsis compared to non-survivors is happening, because the patients which we had studied in the previous part of the work were all all males. And we know now that males have lower basal levels of haptoglobin. Hence, we wanted to know if there is any role of the steroid hormones or the sex hormones in modulating this immune response in patients. So since instead of using patients, we used healthy human volunteers to draw their blood and use them as an ex vivo model. Next, using this model, what we have done is we have evaluated the role of sex hormones in regulating LPS-induced response in human whole blood samples. This was basically done to find any important links between the gonadal hormones and the immune system, steroid immunosuppressive. Whereas Female gonadal steroids increase activity of humoral response. And of course, as I said, the incidence of sepsis was much higher in males compared with females. A previous study led by Venu et al. In 2013 had also reported a significantly higher incidence of bacteremia in males compared with females. And therefore, we continued this study using the ex vivo human blood model. And we found that in female blood exposed to, in female blood in general, the haptoglobin levels were higher compared to males. And when these blood samples were treated with LPS, the inflammatory response was higher in males compared to females. And a higher expression of HP or haptoglobin in males, in females than males. 1 second. Later, what we have done is we have used healthy volunteer blood samples to expose them and treated them with estrogen and testosterone. So we added extra estrogen to female blood and extra testosterone to male blood, simply because the basal levels of testosterone and estrogen vary with females. We wanted to find out if estrogen and testosterone as such have any effect on this system. So when we expose female blood to LPS and female and male blood to LPS and respective sex hormones, we found that in males the finding that inflammatory cytokines are higher were confirmed, and haptoglobin levels were lower on exposure to testosterone, whereas in females, exposure to estrogen increased the basal haptoglobin levels. So this indicates that the gender can to some extent affect the inflammatory response in blood. And the same we observed that by Western blotting, where the protein levels of haptoglobin were higher in females compared to males upon exposure to estrogen and testosterone in respective blood samples. Therefore, we could observe that haptoglobin has some association with estrogen. Also, it could be because the basal levels of haptoglobin itself are higher in females, and increased haptoglobin level is inversely proportional to the TNF-alpha, which is an inflammatory cytokine expression. And high haptoglobin levels in blood may be due to estrogen, which is one of the factors which could be contributing to protection against infection. Then the next question we wondered was, is haptoglobin in any way helping the cells to tolerate the amount of toxin which might be released in sepsis by the causative or etiological pathogen? So to investigate this, before we understand, it is important to investigate previously documented effects of haptoglobin downregulation, which causes abrupt increase in pro-inflammatory cytokines. And here the goal was to basically study the effect of haptoglobin in regulating the inflammatory cytokine levels in ex vivo model of endotoxin tolerance. For this model, what we have done is that we have primed cells. Okay, we have primed THP1 macrophage cells with small amounts of LPS. And the cell supernatant, the medium we have taken, and we have put on unprimed cells. Okay, and this unprimed cells would be rich in haptoglobin, and haptoglobin if possible, right? And in this model, what we observed is the initial exposure to low doses of endotoxin causes cellular reprogramming towards an anti-inflammatory state, in which what is happening is the TNF-alpha production by monocytes like THP1, which initiate into macrophages and decreases in response. Therefore, this is indirectly promoting endotoxin tolerance. And this was not observed in cells where we have added haptoglobin antibody, indicating that haptoglobin must be available free to promote this endotoxin tolerance state, which is reversed by the antibody. Higher when there is no anti-HP neutralizing antibody, which actually results in reversal of endotoxin. And basic findings of this study is that LPS primed PBMCs or the blood cells which were used in the subsequent the confirmation of the pH. The results correlated with endotoxin tolerance, and the observations were in agreement with previous work on effect of haptoglobin on pro-inflammatory cytokines. And basically, we are saying here that neutralization of haptoglobin showed reversal of endotoxin tolerance, whereas blocking it in conditioned media showed increased inflammatory cytokine production. So we were wondering if this has any effect, and basically haptoglobin is a liver protein. It is secreted by the liver. Any conditions which cause liver dysfunction will cause haptoglobin non-secretion or some defects or dysfunction in haptoglobin secretion. Haptoglobin, and we were wondering if this is also true in sepsis. If there is any effect on haptoglobin secretion. Now, since we could not use a human volunteer, what we have done is again an in vitro and ex vivo model. And PKC delta is also known to be reported compound in a plant called Kamala plant. It is widely used in research, and several studies have shown that inhibition of PKC delta by rottlerin in haptoglobin transfected overexpressing cells like THP1, they reduce haptoglobin secretion into the medium. And another study also suggested that by Mi Kyung O et al, it also suggested that haptoglobin is produced in hematic cells and retained within such cells and is finally secreted at specific inflammatory sites when they are activated by stimulants that transduce PKC delta signals such as LPS, right? So we observed that in cells treated, PBMCs, when they were treated with not haptoglobin, but when they were treated with LPS, and when serum was separated, and we performed an agar-well assay with these sera obtained from the blood samples treated with LPS, we observed that when we add LPS only, haptoglobin is probably secreted, because in agar-well assay you can see a zone of inhibition marked by red and blue arrows. Okay, but when we add this rottlerin to the same samples before treatment, we observed that this zone is absent. Okay, and also when we add haptoglobin antibody also, this zone of inhibition is not present, indicating that haptoglobin is one of the major antimicrobial proteins, because when you add antibody against haptoglobin, this zone of inhibition is not seen, whereas when there is no rottlerin or haptoglobin antibody added, there is a zone of inhibition, indicating that haptoglobin is partially inhibited by PKC delta inhibitor rottlerin, which is shown by complete restoration of gram-negative and partially gram-positive bacterial growth on blocking of haptoglobin. So the same thing, the same serum when we have used on a spread plate method, we have seen the same thing. When LPS is added along with rotlerin, there is no inhibition of bacterial growth. As you can see in the colony count assay, you can see the pictures of the plates. Whereas when when only LPS is added, the bacterial growth is completely prevented. And also, upon addition of PKC delta, the inhibitor of PKC delta, which is rotlerin, when we add it to whole blood and expose it to LPS, with increasing concentration of LPS, you can see increasing haptoglobin concentration, but this is inhibited when rotlerin is added. Therefore, the culture supernatants from the PBMCs obtained from human blood cells showed decreased secretion of haptoglobin on rotlerin pretreatment in presence of LPS. Therefore, we understand that PKC delta is one of the important factors in secretion of haptoglobin and its activity. So on the whole, we are when we present our findings, we are when we summarize our findings from these parts of this work on sepsis, we are finding that delayed acute phase response is the major factor because of which the risk for severity of sepsis may be increasing, specifically in male patients, and this may be contributed due to the lack of estrogen and decreased low expression or the decreased expression of haptoglobin in early stages of sepsis may be the contributing factor to impaired acute phase response. And also that higher haptoglobin levels because of estrogen may be having some anti-inflammatory role, and haptoglobin was also identified in this study as one of the major antimicrobial peptides secreted in the blood in response to LPS, which can be blocked by the factors which inhibit PKC delta, meaning that haptoglobin secretion is dependent on PKC delta for secretion in blood. And another thing is that endotoxin tolerance study showed haptoglobin regulates immune tolerance, where it is shown to regulate TNF alpha gene expression via a positive feedback mechanism, indicating its anti-inflammatory role. And as I said previously, higher incidence of sepsis may be due to paucity or lack of estrogen compared with females. Therefore, haptoglobin may also have a gender-specific role in regulating immune response and may be a potential marker in sepsis prognosis to predict possible mortality. And from this study, we were wondering if in any way haptoglobin diagnosis in patients for sepsis can help us diagnose the risk of severity of sepsis in such patients. So as I had mentioned in the beginning, the current diagnostics of sepsis are dependent on PCR-based pathogen detection also, like C-reactive protein also, procalcitonin, which is a gold standard, then SOFA scores, which are taken by the doctor depending on the patient conditions, like blood pressure, temperature, and so on. And some new markers are lipopolysaccharide binding protein, which is a non-specific marker for inflammation, STREM1, STREM1, which is also a marker for inflammation but not sepsis, and SUPAR, which is having a low diagnostic value for sepsis. So we were wondering if, because of these limitations, if we could do anything for our diagnostics of sepsis, especially because current diagnostics are quite expensive by Indian standards and are time consuming. And in several cases, the onset to mortality in sepsis is very short, like from 0 to 48 hours. Sometimes within 24 hours, a patient may meet with mortality due to the lack of balancing of the immune response. And as I have mentioned previously, haptoglobin is elevated in sepsis patients with better outcome who recover easily. whereas its levels are lower in people who do not survive and unable to manage sepsis. And we were thinking that haptoglobin may hence serve to predict the severity-based immune status of the patient, and we wanted to test this hypothesis. And we were thinking that together with procalcitonin, which is the routine marker for sepsis diagnostics, can be utilized to predict severity of the condition in patients. For this, we have designed in our study a test called as SEPSURE, for which we have received a grant recently, which is just under sanction, which will be sanctioned very soon. And the objective of our diagnostic test is make the diagnosis of sepsis affordable, quick, and accessible. And most of the current diagnostics are expensive, as I said, require costly and high-end instruments. They delay treatment due to their duration of diagnosis. They are non-specific. And because of this, it is quite traumatic to the patient, because at present, with these diagnostic methods, we cannot know the severity risk of the patient. Therefore, we have targeted the test for severity for the risk of sepsis, for severe sepsis risk. Okay, and we wanted to make it rapid and point of care, which can be done at the patient bedside. Excuse me. And this may help in early prediction of severity along with diagnosis of sepsis, and also maybe help the doctor in better patient management. The principle of this test is that to detect procalcitonin and haptoglobin rapidly using a gold nanoparticle based diagnostic test. Okay, and we observed from several studies that the haptoglobin protein as such is differentially glycosylated in disease conditions like cancer, hepatitis, and so on. So we were wondering if the same thing is happening in sepsis, and is it affecting recovery and mortality. Therefore, we collected the plasma or serum of the patient, and we designed the test where we used certain lectins to bind to gold nanoparticles in one set, and in another set, we bound haptoglobin antibody to the gold nanoparticles. And we conducted this like a simple dot blot on a nitrocellulose membrane, where we have spotted the serum of the patient, okay, up to just 1 to 2 microliters on the strip, on the nitrocellulose strip, as you can see here. Okay, and we have added the coated gold nanoparticles to this serum one by one. First, we add haptoglobin antibody, and second, we add the lectin bound to the gold nanoparticles. These lectins are specific to fucosylated residues of proteins. I'm not going into detail here about the chemistry and biochemistry of this assay, but simply what the strategy was to find haptoglobin and specifically bind its sugar residues attached to the haptoglobin protein. So due to addition of 2 types of nanoparticles, this has aggregated the gold nanoparticles, and the test was observed as a reddish to brown spot in positive cases. And this we have confirmed using survivors and non-survivor samples. As you can see in panel A and B, when we have done the test for procalcitonin in the same way, where we have bound gold nanoparticles to procalcitonin and detected procalcitonin in the serum or plasma sample of the blood, both survivors and non-survivors showed the binding. This is mainly because in healthy donors, HD1 as you can see and HD2, in healthy donors procalcitonin levels are low, very, very low to absent. Whereas in sepsis, it increases in any case, survivors or non-survivors. So from this test, it is clear that because of the binding of lectins to gold nanoparticles and also haptoglobin antibody, this test became specific wherever haptoglobin was fucosylated and its levels were high, and therefore high number of fucosylated residues, this spot resulted only in survivors and not non-survivors. In non-survivors, only procalcitonin window is showing red to brown spots. That is the third and fourth lane in the bottom rows. So this says that the test we have made is specific for procalcitonin and also fucosylated haptoglobin, and this could probably differentiate the risk of severity in patient, and maybe we can point out if the patient is having a risk for severity, and therefore the patient can be managed more effectively by the doctor. The importance of this test is that it was done within 30 minutes. It required less than 10 microliters of patient serum. Since this fucosylation is a salient feature here, from early stages itself, it was observed that the fucosylation is quite high and it could be detected by the dot blot, and we did not require any high-end equipment for this. This result is visible with the naked eye by the aggregation of gold nanoparticles. And because of this, we do not require any specialized labor. Any person who can handle a micropipette and know what is serum and plasma and how to use an append-off and all can easily perform this test, and we could generate minimum bio-waste by this method. And why we have devised this test is, especially in India, because of our 1.3 billion population and very high incidence of sepsis in the ICU, there is a very high demand for rapid diagnostics of sepsis. At least $200 million right now, and within next 5 years, it is going to increase by at least 1/3. And because there is a lack of high-end diagnostic facilities in rural areas where 80% of the population resides, using this point of care method of diagnosis may definitely help for a better diagnostic method in such areas. And as I mentioned previously, the diagnostics of sepsis take a lot of time. And if we can shorten the test for within minutes, to just minutes instead of hours, do it at patient bedside, definitely we can target all the ICU patients and make sure that from early onset itself to manage the patients. So this test was recently patented, and we were awarded with this patent. And this is our lab. The girl wearing the blue dress at the back in the left photograph is the one who has worked in the early studies on sepsis with endotoxin tolerance, 2D electrophoresis and all. And the girl wearing the checked kurta, Sandhya. First girl is Swati, and my second PhD student, Sandhya, has worked on the diagnostics of sepsis. And the rest are working in similar areas. And finally, I would like to acknowledge the Indo-US Science and Technology Forum and UGC DRS SAP1, FIST1, and Basic Science Research Support provided at the Department of Microbiology, and our former professor, Surya Satyanarayan Singh, who was the director of DBT SLAIR in Osmania University, and our collaborator, Dr. Venkatraman Sridharan from Global Hospitals, Hyderabad. With all their help, we could do this work and successfully find something interesting about sepsis and its diagnostics. Thank you.
Thank you, ma'am. Thank you for the wonderful and informative lecture.
Thank you. Thank you for inviting me.
Thank you for accepting our invitation, ma'am. It was wonderful to have your presence here as a keynote speaker. Thank you very much, ma'am.
Thank you so much.
On that note, we are wrapping up the keynote presentations for this conference. And following this, we will have the technical sessions, that is the paper presentations. We are a little ahead of schedule, which is good. So in a few minutes, we will take a short 5-minute break, and after that, technical sessions will begin. And a few announcements for the participants is that your oral presentation time limit is a total of 10 minutes. of which 8 minutes is for your presentation and 2 minutes is for any question and answers that the session chair might have. For poster presentation, it is 4 minutes for presentation and 1 minute for the discussion. And in the agenda, if your name is under the main hall category, you would have to remain in the same room. If in your agenda it says hall A, you don't have to do anything. We will transfer you to that particular hall. If you have any queries, you can always let me know. You can call or WhatsApp me as well. In 5 minutes, we'll be allocated with your specific rooms, and we will begin the session shortly. Hi again. Those who have your presentations in Hall A, we have added you to that particular hall. So if you see any pop-up in your screen asking you to join a particular breakout room, please accept that because that is where your presentation will be. So you cannot present in this room, your name will be in Hall A. So please make sure that you have joined your breakout room. We have added all the present participants to join that particular room. So people who have not yet accepted the request, please join. Hi, participants. Just a few more minutes. The session chair is about to join. So as soon as we have the session chair, we will start the session and we'll go according to the agenda. Thank you so much for your patience. Hello, ma'am. Good morning, Dr. Swati.
Good morning. Good morning.
Good morning, ma'am. With your permission, may we start the technical session, ma'am?
Sure. Please go ahead.
Thank you, ma'am. To begin this technical session, I would like to first thank and welcome Dr. Swati Kumari from Sri Ramachandra Medical College for accepting to be the session chair and helping us with this conference. Thank you so much, ma'am.
My pleasure.
And I would like to welcome all the participants who have joined here for your presentation, and we wish you all the best for your presentation. Just a few reminders before we start. Please make sure to stick to your time allotted. If you have given an oral presentation, your presentation is for 8 minutes, followed by 2 minutes for any questions if the session chair has. And poster presentations are for 5 minutes in total, 4 minutes for the presentation and 1 minute for the discussion. So since you will may be already aware that we provide award certificates for selected participants, so give it your best shot and definitely do adhere to the guidelines and the time limit, so that you could get your highest points for that. Thank you so much, everyone. So we'll start with the first presentation. I invite Dr. Saransh Srivatsava for the oral presentation.
Before they begin, I just
Yes,
want
ma'am.
to say all the very best to all the participants. And I will just echo what ma'am has said, that please stick to your time, don't extend the time, and be relaxed and prepare and speak well. So anybody will be there to manage their time?
I can do it, ma'am. I can do it. I'll be here.
Please go ahead.
Good morning, ma'am. Thank you so much, BioLEAGUE, for giving me the platform to present my paper presentation. Okay, I'm sharing the screen for my today's topic. I hope you guys are able to see my screen.
It says started. Oh yeah, we can see the screen.
Okay, so my topic for the today is role of salivary biomarkers in the periodontal disease. Myself is Dr. Saransh. I'm an assistant professor in Sharda University. So starting with the introduction. So a biomarker is a substantial material that exhibits a biological state and is used as a goal measure to evaluate the current and the potential future course of a disease. A substance is characterized as being impartially estimated and evaluated as a sign of usual biological cycles, pathogenic cycles, or pharmacological responses to a therapeutic involvement. Biomarkers for periodontal health and disease by finding out and using several natural media, including saliva, serum, and GCA. The mobility and severity of the periodontal infection can't be projected by a sole biomarker. In order to predict how a disease may manifest, combination of biomarkers are used. In past, radiography and clinical tests such as clinical attachment level, bleeding on probing, and probing pocket depth were used to diagnose periodontitis. However, because saliva contains both host and bacteria-derived molecules that express inflammation, and for the analysis, development, and the response to the therapy of the periodontal problems, both soft and hard tissues, which can be employed as a biomarkers. So the pathophysiology of the disease is tied to the potential biomarkers for the periodontal disease. Thus, each one is influenced by infection, inflammation, and the degeneration of the supporting tissues. So these are certain classification of biomarkers which are classified into proteomic, genetic, microbial biomarkers, and other biomarkers. So what is the research problem? So in periodontal research, where ideal standards have not been explained, the data assessing the action required for periodontal disorders have been a dubious use and are not equivalent. So the norms for the periodontitis analysis extend from 3 mm to 6 mm in pocket probing depth and from 2 mm to 6 mm in clinical attachment loss, which is a sign of the periodontitis. These measurements were made at various sites using various measurement techniques in the studies that were reviewed. Periodontal disease cannot typically be evaluated radiographically for attachment loss or alveolar disintegration. Additionally, radiographs can only depict 2D image of a 3D scenario. The majority of the indexes report historical data on past disease rather than the actual occurrence of a disease. Additionally, rare periodontal markers assess the distinct scientific traits or factors. The issue of the clustering of data which arise when several scores are gathered for a single subject and has a significant analytical ramification. Fifth one, the clinical measurements are almost often gathered by multiple examiners in epidemiological research, levitation the question of inter-examiner measurements. So the solution was a diagnostic marker tracks the disease occurrence and development. Saliva, GCO, that is gingival clavicular fluid, serum, and the mucosal transudate are samples utilized meant for the identification of biomarkers in periodontal health and disease. Interpretation for any disparity among part mouth or full mouth valuation or correction factor, which should be determined if a partial mouth assessments are conducted. Third one, the examiner should be subject to strict training and calibration requirement. And the details of this drill and the standardization should be clearly disclosed. So here I'm concluding my topic. The realm of the identifying the oral disorders, there has been a substantial uptick in the development of tools to monitor the periodontal. The evaluation of disease has sparked the development of novel therapeutic techniques, many of which are focused on host modulation, as well as the identification of numerous new biomarkers. Further on, the research should be concerned on the creation of markers because no one indicators can meet all the criteria required to assess the clinical state of the periodontium. So, what are the scope for further work? And the basis of this, saliva is a biofluid potentially rich in diagnostic indicator for both oral and systemic disorders. In recent years, numerous methodologies have emerged for evaluating the microbial and molecular constituency of saliva. Unique saliva-based biomarker profiles can be correlated to certain disease and may provide critical information regarding an individual's current physiological state. Discovering, validating, and understanding saliva-based biomarkers could have a considerable role in establishing oral fluid as a credible diagnostic biofluid. The field of saliva diagnostic indeed has a great translation and the clinical potentials. So these are my references. Thank you so much.
Nice presentation, Mr. Sharans.
Thank you so much, ma'am.
I have few questions to ask from you.
Yes, ma'am.
When you make any presentation, try to make in a slide maximum 7 to 8 lines.
Okay, definitely. I'll do that.
And you should cut short the words.
Okay, ma'am.
Let it be self-explanatory, or you can add your own words.
Okay, ma'am.
And research problem, when you mention, it's better to restrict to few of the research problem than mentioning a lot of research problem.
Okay, ma'am.
So have you checked any periodontitis marker in your patient which you have seen?
Yes, ma'am. Basically, in my previous studies, we usually use alkaline phosphatide and interleukin-1 beta to evaluate the condition of the periodontitis in the patients. As these markers are normally present in our body, but at the time of the inflammation, basically these markers get increased, which will indicate what the severity of the disease will occur in future, so that we can plan the treatment according to that particular, on the basis of that particular markers like alkaline phosphatase, interleukin-1 beta. In future, I'm planning to do the studies also on the spartate aminotransferase, ma'am, and the cathetase, which I'm going for the planning for the doing this using 2 markers on the basis of this also, and even also using the interleukin-1 beta in the cases of diabetes mellitus cases, in the case of periodontitis.
So you have seen those markers in a selected immunocompromised like diabetic patient or normal healthy patient presenting with inflammation?
Ma'am, basically, definitely, if the immunocompromised patient like diabetes, the marker will be increased definitely. But at what level the markers are increasing and what level they are affecting our oral cavity in the on the remaining teeth, the level of the bones on that particular area, according to which we have to plan the treatment according to particular markers.
Okay, thank you.
Thank you so much, ma'am.
Thank you very much for your presentation, sir. For the next presentation, I invite S. Navneeta Kumar from Day2 University for the oral presentation.
Hello, everyone. Am I audible?
Yes.
Yeah, yes. Is my presentation visual? Hello?
Yes, sir, it's
Uh,
visible.
yeah, this is. Good morning, everyone. This is Navneet Kumar from the Gautam University. So today I'm going to present my PPT. I work with endophytic fungi, and from I collected endophytic fungi from the Eastern Ghats. So here my topic is evaluating phytophytogenic effect and check its growth promotions against several pathogens of maize in several pathogens against maize. We all know like all world is going through the organic, and every one in the world are preferring organic rather than using chemicals, pesticides, and everything. Maize is one of the stable crop in the world which needs the focus because we know like maize is produced in a large quantity and they use a lot of pesticides and chemicals to produce it. Here what I did, I collected many species from endophytic fungi from the Eastern Ghat area. Eastern Ghat is having a really importance in the if you take the climatic conditions or the geographically, it is having a really good importance in India. So here my aims to collect potential strains from the inner parts of the Eastern Ghats and to evaluate the antagonism against the pathogens which are agriculturally important, and to extract the secondary metabolite from it, and to check the bioactivity of the same compound and check the extend the antimicrobial activities of this same thing. And later on, I also did the field studies. So here my materials and methods. I'm going to the results directly. So here you can see the areas where I had collected all the samples. Most of them are from the Eastern Ghat area and situated both Chhattisgarh, Odisha, and Andhra Pradesh, as well as some part of Telangana. As well as I have taken some references from several parts, which included Western Ghats also, but the samples are very less, and just those are just for sake of comparison. And here we go. So after taking the samples, here we can see the different fungal samples in the right hand side. And after doing the sample, first thing is identification. Identification was done using morphologically. Morphological, I did the identification, and the strains which are showing the more compatibility and the showing more antagonism, I have taken for the ITS sequencing. Here the ITS sequencing showing the Acrolinema vagum. I'm presenting one of my strains actually. So after doing the ITS sequencing, I have went through the blast sequence, and after blast I have considered the phylogenetic tree. Here we can see the first antagonism of the strains. Here you can see it is growing well against the few of the major pathogens of maize, which is first is Persicum, second is post-harvested. We got aphlogenic, we got Aspergillus flavus infection in generally in maize, which cause aflatoxin in the maize, and that is a class 1 carcinogen, which is which will lead to the HCC in patients. So here I've taken the 4 strains, which include major the Persicum and the Aspergillus flavus. So after doing it, it is the percent of inhibition against the several pathogens. You can see it showed 80% inhibition of the growth in case of Persicum, and as well as 20 around 80 70 75% in case of Aspergillus flavus in the on the plate actually in vivo. But again, I have taken Aspergillus niger and Fusarium. When it comes to Fusarium, it shows around 40% only. But Fusarium is not the top pathogen in the maize. So here next I did the zone of inhibition test using MICs and all. And here I have optimized the culture using several conditions. Here the dextrose came as the top carbon source, and the nitrogen source is petcone. Whereas the temperature is around 27 degrees Celsius and the pH is around 4.5. After doing it, I have extracted the metabolites. After extraction of the metabolite, I went for GCMS. In the GCMS, you can see several compounds which are producing anti-fungal activity as well as anti-cancer, and some of it is also producing some antioxidants. So here the compound which are already proven, like already it is there in the literature. So I have taken it. And some compound which I have extracted also, now I'm working on doing crystallization of the compound and finding the novel compound also. And here the oxygen count is around 2.20. These are the phytochemicals, and flavonoids and phenolics also I did it. These are all the antioxidants which can be produced in it. And here I did the root and shoot length, like a germination test. I did it. In the germination test, I have did the root and shoot length of taken the root and shoot length of the particular seeds. And here you can see comparing control, it is 2 centimeter more in case of my culture. Root length is also around 2 centimeter more than the control. And here you can see the first one is the test one. The field studies are still going on, so that I can't produce the whole thing. I'm just giving the glance of it. That the control one is really grown well, and the test one is really grown well when comparing to the other controls. So here we can see that this is having a good potential to being a like biopesticide. And the summary, though, so I will conclude it here. So the thing is, I can say this is no one have investigated against the like Acroleimma vegum. It is a saprophytic fungi, but in the literature there is no presence of biopesticide activity of the current strain. So further other investigations as well as the dye production and industrial application can be found for this for the current strain. Thank you very much.
Thank you, Mr. Navneet. Extensive work, very nice, very good.
Thank you.
Same suggestion as I gave you a previous participant. The content in a slide should be less. And
yes.
I just want to go to your phylogenetic analysis slide, which you projected.
Oh, one moment.
Yeah.
Yes, on screen. Yes, ma'am.
So your isolated strain is the first one, right?
No, ma'am. Actually, I have isolated few of the Acroleimma vegum actually. So here I'm projecting one, and it is the most like in between those strains. No, it is producing the more one. Like it is having a more aggressive actually. So I have taken the phylogenetic analysis of all. Actually, it is not the particular one I have taken it.
Okay. So what software you use for this?
I have used Mega X, ma'am.
Okay, so that should be mentioned in your slide.
Yes, ma'am. Yes, ma'am.
What was your bootstrap value? Which software you used?
Yes, ma'am. Yes, ma'am.
And
bootstrap around, yes, ma'am. Yes, it was the 1000, and I have using neighborhood joining method, actually.
Okay. And your isolated strain picture, which you plate picture. I have hope it is Sabzrot dextrose agar. Which agar?
Yes, no, it is PDMM actually. In several dextrose agar, it is not growing well. That's why I have optimized it into PD actually. PD is growing well, and we have to decrease the pH a bit less to get the optimized growth actually. That's why I did the culture. I have did the pH and all the tests to see what's the optimum condition to growth actually. Then after doing that, I have concluded like we need to reduce the pH to get the optimum growth actually.
How long these isolate takes to grow on your culture media?
It takes around 7 days, ma'am, to get the fully grow actually. But generally, the mycelium grow in 4 days, and the sporulation start at the 5 to 6 days. But if you say like the optimum growth, it on 7 days.
Okay, so how you prevented the environmental contamination? Did you do anything, or did you face a lot of contamination?
Yes, ma'am. Yes, ma'am. Actually, I have I faced a lot of contamination because it is so much long time that we are keeping the plate outside. So what I do, I use Ziploc bags actually. I sterilize the Ziploc bag in the UV, then I again use them. So after starting using this Ziploc bags technique, I'm not getting any contamination actually. So that's how I prevented the contamination. But in case of liquid culture, we have to seal it with the paraffin. So so that we can't get the culture actually get contaminated.
Okay, thank you. Thank you.
Thank you very much.
Thank you, Mr. Navneet.
Thanks,
One little note for all the participants. Just a gentle reminder for the participants to make sure that you are in the correct hall. If in the agenda your presentation is in the main hall, you are in the right place. If in the agenda your name is under hall A, you have to join the breakout room. We have already assigned, a few of you have not accepted it. So please make sure that you are in room 1 because you cannot present here. Your name will be called in that room. So please make sure that you are accepting the invitation to join room 1. Thank you so much. And for the next presentation, I invite Ms. Bhagat Sashi Kumar Parmeshwar.
Yes, ma'am.
Hi, please share your screen.
Yes. Is it visible, ma'am?
Yes, it's visible, ma'am. Your voice seems a little low. I don't know if it's only for me.
No, it's slow.
Oh, okay. Could you speak up a little bit?
Yeah, ma'am. Yeah.
Okay. Okay. Yeah. Thank you.
Is it good now?
Yes,
Yes.
yes, it's a lot better. Yeah.
Good morning, ma'am, and good morning, everyone. I am Bhagat Sashi Kumari Parmeshwar, PhD scholar under Professor Ashish Sachan from Department of Life Science, Central University, Jharkhand, and I am today here to present and give an oral review of on the topic lipopeptide classification mechanism, synthesis, sustainable production, and application. So, moving on to introduction. As we know, there is antimicrobial resistance nowadays, and it was recorded that 1.27 million deaths were caused due to infection caused by bacterial antimicrobial resistant microorganisms. As we know that the drugs are getting resistant to the microorganism, there is a need to find a new drug. To find new drug, but it is but as it is time taking, so we need to work on the existing
Please
drug,
change your slide.
which
Please move your slide. Yes,
yes,
and
ma'am.
come closer to your system, please.
Yes, ma'am. And so developing new drug and commercializing can take up to years and years. And so working on the existing drugs like modifying enhancement in mechanism can be solution for it. One of the such molecule is antimicrobial peptides because there is less known resistance for such antimicrobial peptide, and it is a part of every organism as a innate immune system part, acting as a first line of defense against different microbes like bacteria, fungi, virus, and parasites. There are different antimicrobial peptides as defensin, dermicin, but one of the distinguished class is lipopeptide, which can also be referred as biosurfactant. Structurally, lipopeptide have two main parts as a peptide chain and a lipid tail. Peptide chain is hydrophilic in nature, and it can be linear or circular in the structure, while the lipid tail is hydrophobic and it is linear in structure. These are some of the lipopeptide structures such as surfactant, lichensine, iturin, and fungicin. As these molecules are amphiphilic, this can self-assemble into different structures as fibrils, micelles, and etc. And one of the useful lipopeptide is daptomycin, which is used as a treating since 2003. Moving on to classification, this lipopeptide can be classified into 5 classes as surfactant, iturin, fungicin, crutaxin, and lolisomycin. Surfactant was discovered in 1968, and it is one of the great biosurfactant, as it can reduce the surface tension of water to 20, from 72 to 27 meter Newton per meter. It is a heptapeptide which is linked to 12 to 16 carbon long beta hydroxy fatty acid chain. The operon consists of 4 ORF as SRF A, B, and A.
145 వరకు ఇక్కడే ఉన్నాను ల్యాబ్ లోనే.
The antifungal, antibacterial, antifungal, antibiofilm, larvicidal, and biosurfactant properties can be seen of surfactant. It mainly attacks the lipid membrane and interfere with the integrity of membrane, leading to the leakage and disruption of cell structure. It includes variants like lycicin, surfactant, and pumilocydin. It shows antimicrobial against Staphylococcus aureus, porcine parvovirus, fusarium, and candida. The Iturin moving on to Iturin family, it was obtained from the soil of Ituri region of Zaire, that is the Democratic Republic of Congo. It is heptapeptide linked to beta amino fatty acid chain, which length up to 14 to 17 carbon long. The Iturin operon have 4 ORF as ITU A, B, C, and D. The initial triad of the peptide chain is conserved, while the remaining 4 are variable. It have variants as iturin A, C, D, and E, bacillomycin D, F, L, and LC, mycosubstilin, mixicin, substilin, movivacin. It also forms 4 and mainly interact with the sterol component and increase the permeability of K plus ions. It have antimicrobial activity against Rhizopus, Fusarium, and Aspergillus. Next, moving on to fungicin family, it is a decapeptide which is linked to the beta hydroxy fatty acid chain of 13 to 19 carbon long. It has 8 of the amino acids are used in formation of the lactam ring. It have 5 fungicin A have 5 operon as 5 ORF, sorry, as FEN A, B, C, D, and E. Fungicin also have unsaturated fatty acid chain in their structure and exhibit strong antifungal activity against filamentous fungi. It is also used as a biocontrol agent and form the pore as its mechanistic action. It shows antimicrobial activity against Rhizoctonia, Xanthomonas, and Pseudomonas. The next family, crutaxin and losilomycin, were newly discovered. It has the affinity against its own producing cell membranes because it have histidine in its structure. The antimicrobial of crutaxin are against mucor and rhizopus. The losilomycin also have the antimicrobial against Xanthomonas, porcine epidemic diarrhea virus, and it also have lower hemolytic activity. Mechanism. It mainly acts by forming the pore, and it interrupt the lipid structure of the membrane, as it have the hydrophobic structure. So it's the main 2 factors that play role in mechanisms are the number of
Ms. Shashi Kumari, can you move faster? Can you move faster? It's
yes, ma'am. Yes,
yeah.
ma'am.
Can you go to if you
Yes,
have
ma'am.
further results, analysis, anything to show?
Yeah, there's only
Ma'am,
like 2 minutes left.
this
So
is review.
Okay, so you have any interpretation to show us? Conclusion to show us?
Ma'am, it's a general one. Like this is the production.
Yeah,
Like
yeah.
as there is a means lipopeptide have many applications as biosurfactant, and it's also used in hydrocarbon remediation. But the production cost is more approximate. The raw material which is used for the production can cause up to 10 to 30% of the cost. So to reduce the production cost, we are using the sustainable media such as soya bean, rice bran, and here are the tables which have been experimentally used for the production, and it has seen that there is means a considerable amount of production, and it also have reduced the pollution. So it is a benefit to use the waste media for the production. And these are the applications, and as it can be used as biosurfactant, recovery, heavy metal remediation, biocontrol agent, antimicrobial, antifungal, antibiofilm, anti-cancerous, anti-parasitic. It can be also used as anti-oomycetes and mosquitocidal activity. And these are the summary and conclusion. As uh, this can be used as an alternative for for treating the infectious disease caused by the resistant microorganisms, and by using the sustainable production can reduce the cost. And as it have many application, it can also be conjugate with nanoparticles for enhancing or for the synergistic effect of the molecule. And these are the references. Thank you.
Thank you, Ms. Ashi.
Yes, ma'am.
You mentioned something, some product which is used against some viral agent. Can you say it again?
Sorry, ma'am, can you repeat the question?
You had mentioned some product which can have antiviral activity against parvovirus.
Ma'am, the Iturin family shows against activity, ma'am.
Against parvovirus.
Yes, ma'am.
Do you know that the parvovirus doesn't have antiviral? No treatment, no vaccine is given for this viral infection?
Ma'am, there was study which have shown that it is means it is showing effect against parvovirus, ma'am.
Okay. So do you have the reference which you have cited here, Zhu et al. 2019, that reference says that Intruin can be used for parvovirus B19, right?
Yes, ma'am. There is a reference, ma'am. I will share the reference, ma'am. Ma'am, I will share the reference to you, ma'am.
How nanoparticle is going to help?
Ma'am, I have what I am doing is as biosurfactant can act as a capping and reducing agent for the nanoparticle synthesis. I am green synthesizing the nanoparticles by using the supernatant which contains the biosurfactant.
I request all the
Which contains the
participants to be
biosurfactant.
muted
And
except the presenter.
so I have using the means I have not just identified the bacteria as I am means doing study on it. I am
Okay.
using the the supernatant which contains the biosurfactant, and have mixed with the precursors of iron nanoparticles. And I have sent the result for the XRD analysis, and I have not got the results, but I have got the size through zeta sizer. It was like 1.62 nanoparticles. I have got that, ma'am.
Good. Thank you. Thank you, Dr. Ms. Shashri.
Yes, ma'am.
Thank you, ma'am. For the next presentation, I invite Ms. Jalada Patel. Ms. Jalada Patel, are you there? I see the name, but she's not responding.
Yes, ma'am, I'm here.
Okay, please share the screen. Thank you. Following Jalada Patel will be Ms. Aishwarya Bandaru. So I request the upcoming participants to be ready with the presentation. Yes, ma'am, please share the screen.
Yeah. Ma'am, there is some problem in loading. I'm trying it.
Okay, Ms. Aishwarya, are you ready with your presentation? Okay. Okay, Ms. Jalada, we will finish the next presentation and we'll come back to you so that in the meantime you can figure out the issue and keep.
Okay.
Okay. Thank you. Ms. Aishwarya, please share your screen.
Am I audible, ma'am? Can everybody see my screen?
Yeah, we can see your screen. Your voice is a little low. If you can speak a little louder, it will be better.
Yes, ma'am. No, it's fine, ma'am.
It's better, yeah.
Yes, ma'am. Good morning, one and all. Good morning, ma'am. My name is Aishwarya and I'm working under the guidance of Gyan Sunna, ma'am. I would like to share today's topic. It is isolation and pigmentation characterization of mangrove cyanobacteria. And as well, mangroves are a very most important environment as we know, and it is very useful for the protection of many hurricanes and the cyclones from the coastal area. And it also plays a most important role in balancing of rich community as a like balancing of environmental nutritions and also the environment threats and things and cyanobacteria is a
Move the slide,
ma'am,
please.
what happened?
Move the slide. Go to next slide, ma'am.
Yeah, sure, ma'am. Yes. Yeah, I just come to the, as we know, even the mangroves, roots, trunks, and trees, everything is has a rich community of nutrients, and also the most important bacteria which is available and help for the balancing of environment is cyanobacteria. It has a special quality of it produces organic photosynthesis, and it is the most important, it plays a most important role in fixing nitrogen, recycling of nitrogen into the environment. And I've just taken some objectives like identification and collection of samples from natural mangroves and industrial mangroves, like as a comparative study. Because as I was going to the literature review, I found many papers where on this identification and isolation of cyanobacteria, but it's little like in any one environment, like natural environment or an industrial environment. But I found very less papers on this comparative study, like taking the two different environments and doing the research on cyanobacteria. So just pick up that most rare topic. So I just did. And after collection of samples, I just isolated and identification of the pigment profile, where those pigments are most important for the photosynthesis process, and the photochemical characteristics and the rest all comparative study. In my research design, we have taken a natural mangrove area which is located in Kakinada, Korangi Wildlife Sanctuary, Natural Wildlife Sanctuary, and the other sampling site is near HPCL steel plant area. It is HPCL and steel plant area. It's an industrial effluent passing through channel. There the cyanobacterial cultures were collected, and both the samples were brought to the laboratory and isolated in providing the required environment, the light and the light source and the other environment conditions. And also the samples were gone through a microscopic examination just to know the shape and the size, just to get the identification of a culture based on the previous study, which have already identified its shape and sizes things. So based on that microscopic examination, I have found almost 3 types of genera of cyanobacteria based on the microscopic examination. In sampling site 1, I have found Anabina and Nostoc, and sampling site 2, I have found Oscillatorio and Nostoc. And these are the cyanobacterial stains which I have identified based on the photomicroscopic examination. And if I gone through its frequency, how frequent these stains are available in that particular area is when I come to the Vishakhapatnam area, the industrial area, where Anabina found more frequently when compared to the Nostoc. When coming to the Korangi Wildlife Sanctuary, the Oscillatoria found more frequent when compared to the Nostoc. And I've already discussed that the pigment profile like chlorophyll, carotenoids, and phycobiliproteins will play a most important role in cyanobacterial photosynthesis process, the main growth of cyanobacteria. So I've gone through those 3 pigments estimation also to my samples, where it's reported in sampling site 1, Anabina reported chlorophyll, more chlorophyll when compared to the Nostoc. In sampling site 2, Oscillatoria produced more chlorophyll content when compared to the Nostoc. And the same, and again the carotenoids also are performed. Even in that also, Anabina showed more carotenoid concentrations, and when compared to the Nostoc. In sampling site 2 also, Oscillatoria showed more carotenoids when compared to the Nostoc. Phycobiliproteins also estimated in my sampling sites, like my samples collected, even in that also, where it showed a little variations between the Nostoc and the Oscillatorio, and then the Anabina and Nostoc. So I would like to conclude my studies out of the 2 sampling 2 different environments like natural mangroves and the industrial effluent chewing channel areas. I found 3 different cyanobacterial stains, and one common stain. One interesting point I would like to discuss in this work is we found the same stain in 2 different locations, different environments, but showing a vast differences between those pigment profile. Nostoc was found in both the sampling sites, but still it has reported too much difference between the both areas. And even the phycobiliproteins also are differed because as per the previous review I've I saw that if the temperature levels are high, the phycobili will reduce. If the temperature levels are low, the phycobili will increase. So it is a vice versa. So in that area, we can assume that industrial area, it may release some hot water into the nearby water body. So because of that, more concentrations in temperatures, the phycobiliproteins may vary. And the natural area where that is also the same thing. So what I was finally want to conclude is there is a too much number of many uses with this cyanobacteria, as it is also used as a biofertilizers and bioinoculants. These all. So if this cyanobacterial stains were come into the field, it would be helpful as a biofertilizers and bioinoculants. And further study need to be conducted to know the complete details of the cyanobacterial stains which have just which have isolated, and how come I can keep into the environment, and how can I can make into as a useful thing. The further study need to be done now. Thank you so much for giving me this opportunity to be here as a presenter. Thank you so much, ma'am. My references.
Nice presentation, Ms. Aishwarya.
Thank you, ma'am.
I have few questions like
Yes, ma'am, please.
So you have studied in natural environment and industrial environment,
Yes, ma'am.
and you felt like the strain grows better in a natural than your industrial because of hot water flow, right?
Yes, ma'am.
Okay. This cyanobacterial strain has been used earlier as a fertilizer.
Yes, ma'am. There are few studies where they've already used as a fertilizers.
What are the
Because
use
there is a few studies where the cyanobacteria, even it is available in rice fields. There is a paper suggested by Samrina Nas, and ma'am have revealed that Oscillatorio uses as a biofertilizer in rice fields. She made a study on that point as exactly what you are asking me.
So you have also used that and tested, or
no, ma'am. Actually, my study is still going on. I have not gone through till to that point.
no?
Yeah, because I was strictly stick to the point is in that location what stains are available and what is the comparison between that. So my point is still to I was stick till there only yet. So I have to see by adding on a point. But thank you so much for that idea, ma'am. I will definitely keep it add to my work.
Thank you, Mr. Chulya.
Thank you, ma'am.
Thank you, Ms. Aishwarya. Ms. Jalada Patel, is your network issue sorted?
Yeah, I think it's sorted.
Okay.
Sure.
Okay, good morning, everyone. I'm Jalda Patel from PhD College of Arts and Science, affiliated to Bhatia University. I'm doing PhD under my guide, Professor Dr. R. Rajendran. My topic for the presentation is exploring the potential of plant extracts and hydrogel formulations for burn wound infections. And now, first of all, we will discuss what is burn. Usually, burn wounds can be due to fire, electric hazards, and chemical hazards. You can see here that the slide in slide that the burn wound can be from first degree up to the fourth degree. One of the main reasons for delayed burn wound healing is infection. In my research work, polymeric gel with antimicrobial extract from natural resources was investigated. Gels are effective in maintaining moisture level and drug delivery for fast wound healing. Now, going to the objective and aim. The aims, as I have discussed before, the investigation of bioactive properties of botanical extracts and hydrogel compositions for the treatment of burn-associated wound infections. Now, the main objectives are to screen plant extract for antibacterial and antifungal activity, determine MBC for selected plant extracts, further characterizations such as phytochemical analysis, antioxidant activity, FTIR, MGC, antimicrobial analysis of chitosan, and physical characterization of the hydrogen. Now, here in materials and methods, I have taken the plant extracts. I have selected the 5 plants: Rosemary sativinalis, Lavandula agastifolia, Mystica fragrans, Nigella setaga, and Chrysopogon vulgaris. Now, these plants, first I have dried the here for Rosmarinus, I have taken the dried leaves, Lavandula, I have taken the flowers, Myristica fragrans, I have taken the seed, Nigella setaga also seeds, and Chrysopogon vulgaris, the roots are taken. As everyone knows that these all are commercially used in preparing either oil or cosmetics. Now, the pathogenic bacteria I have taken from the PSG hospitals in Coimbatore. They have given me the bacteria which are collected from the burn ward. They all 8 samples. 7 bacteria are collected. These are the names. Now for the methods, the common methods I have first gone through the basic methods and then I have gone through the advanced ones. First, we will go through the antibacterial activity assessment. Here we can observe that the leaves of Rosaminus officinalis shows the highest antibacterial activity. So I have selected it for further testing. Now here in antifungal activity, it can be seen that Myristica fragrans showed higher antifungal activity. So it is selected for further experimentations. Now minimum bactericidal concentration is done and it is found to be in the breakpoint is found to be in between 1250 microgram per ml to 156.25 microgram per ml. Next is minimum fungicidal concentration was observed, and that was found between 8 microgram ml to 16 microgram per ml. Now, ethanolic extract of leaves of Rosaminus officinalis and ethanolic extract of maize of Myristica fragrans had possessed highest antibacterial and antifungal activity respectively, and were further taken in a ratio of 1:1 concentration, and their antibacterial assessment in case of pathogenic bacteria was performed. It was observed through zones of inhibition, formation of end of incubation period. Highest zone of inhibition was formed in case of Klebsiella pneumoniae, and the combinatorial herbal extract possessed antibacterial activity in all the pathogenic bacterial strains. And in the comparison with tetracycline, it was observed that the tetracycline antibacterial activity range is slightly higher than the combinatorial herbal extract. Since the combinatorial herbal extract possesses efficient antibacterial activity against the pathogenic bacterial strains, this proves that if loaded into the hydrogel, it can be used to treat bacterial infections. Now, in here, it was observed that in antifungal activity, it was observed that highest antifungal activity occurred in case of Candida albicans, and on comparison of antifungal activity of herbal extract of ketoconazole, it was observed that ketoconazole exhibited higher antifungal activity against the pathogenic fungal strains. The combinatorial extract was further used for the determination of minimum inhibitory concentration of fungal strains. Now first we will go for the bacterial strains. As mentioned earlier, ethanolic extract of Myristica fragrans possessed highest antifungal and Rosmarinus highest antibacterial. So here you can observe the antibacterial MBC4 antibacterial fungal analysis, and here it is given the MFC4 combinatorial extract of Myristica fragrans and Rosmarinus officinalis. Now, after that confirmation of that, we have gone for phytochemical analysis of both Rosmarinus as well as Myristica fragrans. With both gave good phytochemical analysis results. So we have decided to go for the antioxidant activity. Here an observation of results of antioxidant analysis of ethanolic extract of Rosmarinus officinalis and Myristica fragrans.
Jalada, can you move to a conclusion? Can you go to conclusion slide, result and conclusion? Ma'am, actually my work is under process, still biocompatibility under process. So I have not concluded the results, but I can go through the. Yeah, if you have any summary to say. I can go through the gel preparation parts, ma'am. Okay. Gel preparation result. First of all, I have taken two polymers, chitosan and gelatin, and with chitosan, antimicrobial activity is observed. As we all know that chitosan is antimicrobial in nature. So first of all, 2 concentration of chitosans were taken from 5% and 0.1%, and the 1% showing better results. So we have further used that one for the testings. Now, to confirm this, we have done the swelling ratio of the
further characterization for physical characterization, we have done spelling ratio, dispersion, spelling ratio, and fluid affinity test. In dispersion, it is non-dispersible in nature. So I have taken various concentration of the gelatin with 1% chitosan, and we can see that it is non-dispersible in nature. Now going to the swelling ratio. Here swelling ratio is here it's observed that the 483 566.6 is the highest swelling ratio it observed, and with combinatorial herbal extract loaded hydrogels, the highest ratio is 320%. Just a minute.
Ma'am, I'm sorry to interrupt you. The 8-minute slot is actually over. Can you please try to wrap up your presentation in under a minute?
Okay, even if we graph, we can see that it should become between 2,500 to 5,000, and it shows that now in moisture vapor or transmission rate is also done, and here also we can observe the good results between 2,000 and 5,000. So yeah, the gel can be taken for further analysis. That's it. Thank you.
Ms. Jalada, yeah, thank you for the extensive work which you have shown in your presentation. And I understand it needs a lot of dedication and hard work behind that. So I just have a few questions to ask. Can you hear me?
Yes, ma'am.
So you compare your herbal extract preparation with tetracycline.
Yes.
You compared your herbal extract preparation with ketoconazole antifungal. Why you chose these?
Actually, sulfur diazine can also be used nowadays, but still I want to see whether it will give good results with tetracycline or not, and that's why I have selected tetracycline. So if it is giving good result, then later on I'm going to compare it
So
with.
you recommend to have combination of this hydrogel with antibiotic, right?
Yes, ma'am.
Okay. Okay, I got the point. Okay. Thank you.
Thank you, ma'am. The next presentation is by Dr. Rishaba Verma.
Good morning, ma'am. Am I audible?
Yes, sir, you're audible.
Shall I start sharing the screen now?
Yes, please.
Is it okay, ma'am?
Yes, sir, we can see us.
Shall I show the presentation, ma'am?
Yes, please.
Yeah, okay. Good afternoon, everyone. Myself, Dr. Rishaba Verma P, doing MD postgraduate in microbiology, Sri Ramchandra Institute of Higher Education Research. My case presentation is about the multi-drug resistant neonatal septicemia due to Proteus tiberius in tertiary care hospital, a case report. So the spread of extended spectrum beta-lactam resistant gram-negative bacilli is the most current clinical issue faced in the globally about the antibiotic resistance. The beta-lactam antibiotics are most clinically useful antibiotic in the treatment of several bacterial infection. Many gram-negative bacilli carry a plasmid-mediated ESBL, extended spectrum beta-lactamase, and being successful in proliferating and spreading extended spectrum beta-lactamase resistant through horizontal gene transfers. The CDC, the Center of Disease Control, classifies gram-negative bacilli resistant to extended spectrum beta-lactam as a serious threat. The organism requires consistent monitoring. They possess several limitations to antibiotic therapy under global threat of public health concerns. The genus Proteus of family Enterobacteriaceae was first reported in the year 1980s. the bloodstream infection due to the protease strain is uncommon and has been brought to our knowledge as a little attention. Here's a case report. A 5 days old neonate was admitted in the NICU in view of tachypnea, decreased feeding, and rise in temperature. It was a late preterm baby who was 36 weeks plus 2 days at time of birth. The past history of the child is elective cesarean section was done due to change in the Doppler and early onset of intrauterine growth retardation with the oligohydramnios. On examination of the neonate, its heart rate is 151 per minute, respiratory rate is 52 per minute, oxygen saturation is 94%, rectal temperature is 136.5 degrees Celsius, weight is 1.84 kilogram, length is 43.5 centimeter. The prominent of the forehead of the baby is positive. So the routine investigation was done. The routine blood and the urine analysis was sent for the lab. 2 sets of blood culture was collected aseptically and was sent for analysis. On bacterial mission, the blood sample flagged on the day 3 of incubation and was subjected to gram staining as well as agar plates. In the gram staining, it showed a gram-negative rods. Subsequently, culture plates was put on the blood and the McConkey agar. In view of short for gestational age, torch panel and urine cytomegalovirus was also sent, where both turned to be negative. On continuing of investigation, baby was found to be icteric in the NICU and was started on phototherapy. Echo was done, showed subaortic ventral septal defect of 4 mm. So usually for the neonate, it should be less than 3 mm. For this baby, it is a 4 mm. So they were started on IV diuretics. Due to dysmorphism of the face, since I mentioned earlier about the prominent forehead of the baby, they have done karyotyping testing also, which is found to be normal one. Due to the fever, they also done the lumbar function for the baby and sent for the CSF sample for the culture sensitivity, which is turned to be no growth and no organisms. What is the culture earlier mentioned earlier about the gram-negative base? The colonies on the blood agar showed the gray moist colonies, whereas the colonies on the McConkey agar were non-lactose fermenting and was inconclusive to the biochemical test. So, DNA sequencing was done. The sample was subjected for PCR-based DNA sequencing targeting the sectioners or innate region, which confirmed it to be a protease severus with a gene bank access number OR 793169. The blast data was found of the protease severus strain GS9 16S ribosomal RNA gene, which is found to be a match of 19.49% accurate. At the same time, antibiotic susceptibility testing was done for vaccine, whereas it is shorter to show resistant to all the type of drugs like ampicillin, second and third generation cephalosporin, glycosides, and the carbapenem drugs. So, on the treatment point of view, for receiving the baby, due to distress, the baby was connected with the CPAP and was started on RT feed. After 2 days of CPAP, the baby was shifted to HFNC, high flow room air. Baby was initially off antibiotics and was started on broad antibiotic like injection piperacillin, switched to injection levofloxacin and was shifted to ward and later be discharged. Discussion point of view, early identification will aid in starting appropriate antibiotic for the patient as soon as possible. The multi-drug resistant phenomenon represents a serious medical problem and constitutes a major challenge to the treatment of infectious disease. Symptoms generally resolve without complication in immunocompetent patient, whereas in immunosuppressed patient, like very young child and the neonates, and for the old age people and the patient with the very comorbidities, there is high chance of infection getting prolonged. So we have to do the antibody susceptibility testing as soon as possible. In the conclusion point of view, the multi-drug resistant gram-negative sepsis is a serious life-threatening and challenging globally. In general, patient clinically suspected sepsis. So, on receiving the patient symptoms, the clinician should withdraw the blood and send for the culture as soon as possible. At the same time, he or she should start the broad spectrum antibiotic as soon as possible. On receiving the culture reports, they should taper down the dosage according to the culture first case report of Proteus septicemia in our region. The Proteus has pathogenic potential and is an emerging opportunistic pathogen capable of spreading multiple antibiotic resistance. These were my reference articles. Thank you.
Dr. Rishabh, which are the common organisms which can lead to septicemia, which you have noticed in your hospital setup?
Septicemia, Pseudomonas aeruginosa, ma'am, Acinetobacter pneumoniae, Staphylococcus aureus, Staphylococcus pneumoniae, and Proteus species. Also
So, how you feel like what can be the source of this causative agent in your patient?
the source of infection, ma'am. It can be an hospital-acquired infection also, because sometimes baby was a neonate. Since it was a cesarean section baby, it was kept in the hospital for 3 to 4 days. The symptoms started day 4th of the life of the child. So it can be hospital-acquired infection, or rarely it can be transferred from the mother also. But in this case, the mother doesn't have any symptoms. It can be transferred in the hospital itself, ma'am.
Did you follow up the case?
Yes, ma'am. I followed the case, ma'am.
So what were the
baby was after treatment of the day 5th of illness, the oral antibiotics in form of droplets was given to the child and was discharged in day 7th of illness, ma'am. Day 5, the baby shifted from NICU to the ward. The 2 days, the baby was in the ward and was symptomatically better. And day 7, the baby was shifted to the home with the oral amoxiclavulinic acid as drops, ma'am. Drop format shifted, ma'am.
So do you feel like early diagnosis has saved the child, right?
Yes, ma'am. Early diagnosis has saved the child, ma'am.
Thank you. Thank you, Dr. Rishabh.
Thank you, ma'am.
Thank you, sir. For the next presentation, I invite Ms. Geetu Krishna.
Hello, am I audible?
Yes, ma'am.
Shall I start? Fine.
Oh, yes, please share your screen and start the presentation.
Okay, good morning, everyone. Myself, Geetu Krishna, Assistant Professor from National College affiliated to the University of Kerala. My topic for today's presentation is a review on the exploration of endophytic microorganisms for the synthesis of nanoparticles, a sustainable approach for the bioremediation of pesticides. Pesticides are substances that are used in the agricultural fields for controlling and destroying pests. As of now, biological as well as chemical pesticides are available. In spite of the discovery of the biological pesticides, the demand for chemical pesticides is increasing day by day due to its quick action in controlling the pest. A report in 2021 suggests that in India, of the 293 pesticides registered, 104 pesticides are banned in other nations, but still they are used in our country. Obviously, these chemical pesticides are going to increase our agricultural yield. However, the excessive use of these agrochemicals has adverse effect on human health and our natural ecosystem. In recent years, nanotechnology has emerged as a helping tool for sensing and remediation of these pesticides. It can be used for the detection, degradation, and removal of pesticides. The green synthesis of nanoparticles will surely be a promising approach in the field of bioremediation. Relevance of my topic is Obviously, the pesticides that enhance the agricultural yields, but its use is a major concern today. It has adverse effects on human health. Pesticide residues accumulates in the human body and causes mutation in the chromosome, which is a major cause of carcinoma that is spreading rapidly in our country. About 8 lakh cancer cases are being reported every year in India. Besides carcinoma, it also causes Alzheimer's, renal dysfunction, hepatic necrosis, fertility disorders, osteoporosis, and so on. It also contaminates the soil, causing the soil pollution, and the pesticides may drift into the water bodies, causing the water pollution. And it also emits the hazardous air pollutants and volatile organic compounds, causing the air pollution. Thus, it is a serious threat to the natural ecosystem. So, the potential solution for removing these harmful pesticides is the bioremediation using nanoparticles synthesized from the entophytic microorganisms. So the role of entophytic microorganisms. First of all, the entophytic microorganisms, these are, the term entophyte is derived from the Greek word endo, which means within, and phyte meaning plant. The entophytic microorganisms are those microbes that inhabit the internal parts of the plant without harming the host plant. And the studies reveal the use of these entophytes as a plant growth promoters, biocontrol agents, phosphate solubilizers, and so on. The bioremediation potential of the entophytic microorganisms has tremendous room for the research and development. Thus, entophytes can play a major role in the bioremediation of pesticides. Now, the role of nanoparticles in bioremediation. The nanoparticles are gaining, or nanomaterials are gaining attention in the remediation of pollutants due to their enhanced physical, chemical, and mechanical properties. Nanobioremediation is the removal of the environmental pollutants from the contaminated sites using the nanoparticles that can be formed by bacteria, algae, actinomycetes, fungi, and plants. The green synthesis of nanoparticles is a promising approach to overcome the pesticide pollution, which in turn reduces the soil and water pollution, of course, air pollution also. The nanoparticles are produced by microorganisms either through intracellular or extracellular processes that is based on the location of the enzymatic activity. And they have the ability to convert the metal ions like silver, gold, zinc, copper, even carbon nanotubes, even chitosan. From these, the nanoparticles can be produced with the help of their secondary metabolites and cellular enzymes. So bioremediation using the green nanoparticles that are synthesized from the entophytic microorganism will be a potential solution for the removal of harmful pesticides in a cost-effective manner. So this is a brief outline of how the entophytes can be used in the bio-nano remediation of the pesticide. The first step will be the isolation, identification, and characterization of the entophytic microorganisms from the plants seen in the agricultural lands or industrial waste depositing areas. And from the isolated entophytic organism, the biosynthesis of nanoparticles is being done, followed by the physical characterization using the UV visible spectroscopy. And then comes the main part, that is bioremediation of pesticide. It will be checked and it will be determined by using gas chromatography and mass spectroscopy. So the conclusion is the use of chemical pesticides of the Second World War has tremendously increased in developed and developing countries. The pesticides are not only toxic to humans, but they pose a great threat to the safety of soil, water, and air quality. The long-term persistence of these toxic chemicals is a serious threat to our natural ecosystem and human health. So there is an urgent need for the bioremediation of these toxic chemicals. The green synthesis of nanoparticles will be a promising approach to overcome the pesticide pollution, which in turn reduces the soil and water pollution and thereby helps the entire ecosystem to heal itself. And it provides an eco-friendly, economical, and efficient method for the detoxification of pesticides. These are my references that I have quoted. Thank you.
Thank you, Ms. Ritu.
Thank you.
Nice presentation. I have few queries. How feasible is the application of nanoparticle in our country?
I have just started the work. This is just a review, and I have seen in papers that the nanoparticles are being used, mainly silver nanoparticles, golden nanoparticles. These are being used for this bioremediation process.
So you have literature review from our country or international?
From our country as well as international studies are being cited.
Okay, thank you.
Thank you, Ms. Geetu. For the next presentation, I invite Dr. Munaza Aman. Dr. Munaza, are you there? Munaza, okay. Dr. Gunimala.
Hello.
Yes.
Yes, I'm trying to share the screen.
Yeah, we can see your phone screen. I think we can see it, but it's not yet, slides are not yet visible.
Screen has got shared?
Yeah.
It's not visible for me. Just a second. Is it visible?
Yes, it's visible.
Go to the first slide. Dr. Munaza, go to first slide.
Yes, just a second. A very good afternoon. May I begin?
Yes, please.
A very good afternoon. I'm Dr. Munaza, resident microbiology, here to present a study, a prospective hospital-based study, antimicrobial resistance pattern of the pathogens associated with premature rupture of the membrane in the pregnant women, a study from a tertiary care hospital. A brief introduction about the topic. That preterm birth is a leading cause of the perinatal mortality as well as morbidity worldwide. Asymptomatic or minimally symptomatic infections of the genital tract of the pregnant women are related to the preterm birth, as all of us know, and 60% of such preterm births are preceded by the premature rupture of the membrane. 10% of the perinatal deaths are directly or indirectly attributed to the premature rupture of the membrane. So according to the NICE guidelines, which are followed by most of the gynecologists in the practice, it recommends, unless the antibiotic is not contraindicated, erythromycin 250 milligram per 6 hourly for 10 days should be recommended to such women who are presenting with premature rupture of membrane, that is rupture of the membrane before completed 37th week of the gestational age. And this erythromycin should be continued for 10 days or in the women until the women is established in the labor. And in such women who are allergic to erythromycin or erythromycin is contraindicated in for some other reasons, penicillin should be administered for orally for 10 days. However, without looking for the epidemiological distribution of the causative agents or the resistance pattern, to follow such guidelines is vague. So the aim of the study was set to identify the antimicrobial resistance pattern of the common organisms which are associated or isolated from the pregnant women at the time of the presentation with premature rupture of membrane. Coming to the study design and setting, it was an institution-based prospective study conducted for a period of 6 months at an associate hospital, Government Medical College, Srinagar. The inclusion criteria included the high vaginal swab samples from the pregnant women from the age of 20 to 45 years with the gestational age 28 weeks to 37 weeks, less than 37 weeks. The pregnant women presenting with the chorioamnitis, history of the preterm birth, or with the final diagnosis of the preterm rupture of the membrane. The exclusion criteria included the high vaginal swab from the pregnant women with the gestational age less than 28 weeks or more than 37 weeks, with intrauterine demise, gestational diabetes, chronic hypertension, or pregnancy-induced hypertension, polyhydramnios, or placenta previa, which could also be the reasons for leaking at the time of presentation. So methodology adopted was, under all available aseptic measures, high vaginal swab were collected after taking detailed history and confirmation of gestational age with commercially available collection and transport system. Identification of the isolates recovered were done by conventional methods, the phenotypical method at the microbiology lab, and antimicrobial susceptibility testing was done routinely by the disk diffusion method. Coming to the results of the study, the most isolated organism was Escherichia coli, which made up about 38.6% of the total isolates recovered, followed by Staphylococcus, out of which majority were methicillin-resistant Staphylococcus aureus, and Enterococcus faecalis. Some of the fewer, like 0.4% Proteus vulgaris Candida species, were also recovered. However, the growth of the concern was was no growth of the sterile specimen, for which we could not go further as they were not looked for some of the fastidious organisms, which could be a major reason of the infection of the genital tract. Coming to the susceptibility pattern, the most common organisms like MRSA, MSSA were sensitive to linezolid, and Enterococcus were sensitive to linezolid. 100% of the isolates were sensitive to linezolid and vancomycin. And there were about 20% of the isolates which were vancomycin resistant Enterococcus. And coming to the antibiotics which are actually recommended by the NICE guidelines, that is erythromycin, only 25% of the MRSA, 35% of the MSSA, and none of the Enterococcus species were sensitive to such this antimicrobial. Being most of the isolates, many of the isolates being MRSA, they were about 100% of them were resistant to the beta-lactam inhibitor combinations, cephalosporins and carbapenems as well. Only 62% and 52% of the MSSA were sensitive to tetracyclines. And not surprising, but very important to describe here is MSSA, only 2% of the organisms, the total isolates recovered, were sensitive to the penicillins, which is a recommended antibiotic by the NICE guideline for such cases. So, concluding the study with a remark that empirical antibiotic treatment given for the premature rupture of the membrane patient as per the NICE guidelines can lead to the inadequate treatment, as the causative agents are highly resistant to such antibiotics. And in the resource limited settings where the microbiological evaluation of the amniotic fluid is not feasible, identification of the pathogens in the high vaginal swabs can guide antibiotic therapy in such cases. The limitation of the study was a total of 439 specimens, that is 35.7%, are sterile, which cannot be proven as sterile, as such specimens were not processed for the looking for fastidious organisms like mycoplasma. Chlamydia could also be the reasons of such infections. And this was an institution-based study. As all of us know, epidemiology varies from hospital to hospital, so cannot be considered for other hospitals and geographical areas. These are some of the references which were taken up for making up the study. Thank you so much.
Swati ma'am, are there any questions for the presenter?
Yeah. Thank you, Dr. Munaza.
Yes, ma'am. Thank you. Welcome.
I have few questions for you.
Yes, ma'am.
Why you excluded women less than 28 weeks when you were collecting high vaginal swab?
Ma'am, this was for actually, this was a study in collaboration with the gynecologist. I am myself a microbiologist, and less than 28 weeks, there could be other reasons for there could be other reasons. It is not.
You are saying the reason is from obstetric point of view, right?
obstetric point of view, there could be the
Okay.
reasons. It
Okay.
was just for PR room to be included, it was less than 37 weeks.
So, other fastidious organism that like Streptococcus, Agalactiae and all, you did not isolate?
They were not recovered. We could not recover them.
Okay. On gram stain, there was
think,
suspicion.
Ma'am, there could be the reason they are still in our setting. They are still, whichever isolate, even from the other these specimen also, they are highly sensitive to penicillin. None of them have, we have not recovered any much of the isolates which are penicillin resistant. And as the first line of the treatment, they are already taking
But
up
you said
the
that there were
penicillins.
most of the isolate were after Escherichia coli. Your second most common isolate was methicillin
Staphylococcus.
resistant. So
Yeah,
those all were penicillin resistant, right?
yeah. And among the MRSA, the organism, all of the isolates were penicillin resistant.
So these patients were outpatient or inpatients?
Yeah, ma'am, they were coming to the OPD, gynae OPD. They were mostly outpatients. And the high vaginal swab was taken up when they presented with premature rupture of the membrane.
Thank you. Thank you, Dr. Manasa.
Okay, ma'am.
Thank you. For the next presentation, I invite Dr. Gunimala Chakrabarti. Dr. Gunimala, are you there?
Yes.
Okay.
One second. Am I audible?
Yes, ma'am.
BioLEAGUE, can you just tell me how to do the slideshow in here?
Yes, yes, ma'am. So in the bottom, you have an option to zoom in,
Oh,
zoom out,
yes,
right?
yes, yes.
Yeah, that
Here,
button.
here, here.
Yes.
All right, yeah. Thank you. Okay, a very good afternoon. My name is Gunimala Chakrabarti. I'm an associate professor in Department of Molecular Genetics and Cancer, NITE Research Center. Thank you for giving me the opportunity to present my work in this platform. Today, I will be talking about a sensitive molecular tool for detection of foodborne pathogens. That is basically development of loop-mediated isothermal amplification assay for detection of major two foodborne pathogens, that is Staphylococcus and Salmonella. Foodborne pathogen, one second, causes disease through consumption of contaminated food and water. So there are over 250 reported disease. 250 diseases reported to be caused by bacteria, virus, and parasites. So outbreak of the foodborne pathogens occur around the world, and 2/3 of the encountered outbreaks have been linked with bacteria such as Salmonella and Staphylococcus aureus. LAMP is one such technique. It is a nucleic acid amplification technique where we use an isothermal temperature, and has been regarded as an innovation gene amplification technology, and it has emerged as an alternative to PCR-based methodology in both food safety testing as well as in clinical laboratories. So I will briefly explain about the principle of LAMP. The LAMP amplification is divided into two phases. At the initial phases, we have the forward inner primers, F2 and FIP, which hybridizes the F2C of the template DNA strand, guiding the synthesis of complementary DNA. The LAMP. Pardon? Yes.
That was just a disturbance, ma'am.
And then in the second phase, it is described by the once the formation of the dumbbell structure, this dumbbell structure serves as a template for the cyclic amplification. And the amplification takes place, and you will be able to find several inverted repeats, and which is usually observed by adding a dye or due to the accumulation of magnesium pyrophosphate, you can actually see a white precipitate in the reaction. So a LAMP is also considered as a, compared to PCR and other molecular techniques, it is considered as rapid, cost-effective, specific, sensitive, and very simple, and can be suggested for use in as a point of care tools for detection of any kind of pathogens. So with that background, the aim of the study is to establish or develop a LAMP assay for detection of Staphylococcus aureus and Salmonella species. With the objective, therefore, the objectives are to develop LAMP-based detection assay for Staphylococcus aureus and Salmonella species, and to determine the feasibility by comparing the performance of LAMP assay with the existing molecular methods, and to also determine the feasibility of screening the samples. So to do that, we have to have bacterial DNA. So the ATCC culture for both Salmonella and Staphylococcus, we have used ATCC culture, standard ACCC culture, and the genomic DNA was extracted by using CTAB method. And then second part will be screening of Staphylococcus aureus and presence of these pathogens in food samples, mainly meat and the milk products. So to do that, first thing what you have to do is we have to design the primer for LAMP assay by using online LAMP primer designing tools. Then once we procure the primer, we need to do optimization of LAMP conditions, that is mainly time and temperature at which the reaction will work, and we need to specify the specificity of LAMP assay has to be checked, and sensitivity of the LAMP developed assay has to be considered. So coming to the results, for Salmonella typhimurium, we have targeted INV gene, which is highly conserved across the Salmonella species, and it is located in Salmonella pathogenicity island 1. And for Staphylococcus aureus, we have targeted RC and FAM-A. These are the genes specific to Staphylococcus aureus only. So when for the temperature was optimized, we have set up the reaction from 60 degree to 65, and the time we set up that 15 minutes, 30 minutes, and 60 minutes, and based on the pattern of band formation, and the reaction was optimized as 65 degree for 60 minutes. And for Staphylococcus, since we have two genes, we have similar assays were done. The temperature ranging from 57.5 to 60 degree was set up for the time range of 30 minutes, 40 minutes, 60 minutes, and 80 minutes. And we have found that for ARC-C, 60 degrees centigrade for 80 minutes gives proper band. And for FAM-A, 65 degree for 60 minutes was considered as an optimized time and temperature for Staphylococcus. So further, all the specificity and sensitivity of the assay was conducted at this optimized temperatures and time. So the sensitivity of Salmonella typhimurium was conducted with 10 time dilutions, And this 1, 2, 3, this is the initial DNA, and this is the dilution, and we could detect up to 10 to the power 6, and with the PCR, it could detect up to 10 to the power 5. And specificity of Salmonella, when we check the specificity of Salmonella with other closely related species, it was found to be very specific because the developed LAMP assay could develop amplified only the Salmonella species. So in case of for Staphylococcus aureus, the positive amplifications was seen only in the assay that contained Salmonella aureus, and indicating the high specificity of the developed assay for detection of Staphylococcus aureus.
Ma'am, I'm sorry to interrupt you. Ma'am, the 8 minutes is about to run out, ma'am. Kindly wrap up the presentation in 1 minute.
Okay, fine. And sensitivity, we have spiked the chicken meat, and with the different concentration of the cultures, and in that also sensitivity was LAMP assay was found to be more sensitive than PCR. So these are we have screened some the samples, meat samples and dairy milk products, and this is a comparison. LAMP assay was found to be more sensitive compared to PCR here. To conclude, study was designed to showcase the application of LAMP technique for detection of this two important foodborne pathogen, and sensitivity of the assay was found to be 100-fold higher than conventional PCR. The specificity of the assay, which is an important criterion while developing a technique for accurate detection of the pathogen, was supported in this assay. And therefore, the assay could be further developed as a point of care device applied in diagnostics and surveillance in industry, for industry and farm cultures, so that the bacterium carrying the meat can be segregated before it reaches the consumers, and screening of suspected samples, particularly during the outbreaks, can be done. So this was part of the DST Women Scientists project given to me. So I would like to thank DST, Government of India, and I would also like to thank Professor Indrani Karuna Sagar, who is my mentor. And I would like to thank Nitte University for providing all the facilities for conducting this research. Thank you very much.
Thank you, Ms. Guniwala. Nice presentation.
Thank you.
I have few questions.
Yes,
Why
ma'am.
did you choose Staph aureus and Salmonella?
Because, ma'am, most of the foodborne related disease outbreak, this two are the major pathogens associated with that. That is why we thought we will. There are other pathogens also being targeted. We are still working in my department. But at the moment, we have completed for Staphylococcus aureus and Salmonella.
So your Staphylococcus is free-form toxin. You must be aware, right?
Yes, ma'am.
It's not released. It's already formed in the food substance. So you chose one which has free-form toxin and one which gets released through food. It gets released in the intestine, right?
Yes, what we have done in this work is, ma'am, we did not, thank you for the suggestion. We did not consider about the toxin part. We are focusing on the detection of this pathogen by this LAMP assay. So it is just a development, and further application when we, in future work, we can consider what you have suggested. Thank you very much.
Thank you.
Thank you, Dr. Gunimala. The next presentation, I invite Ms. Donia Joseph.
Yes. Can I share the screen? Am I audible?
You're audible, ma'am. Please share the screen.
Okay. How can I? Is the screen visible now?
Yes, it's visible. PPT is not yet opened, but we can see the folder.
Okay. Is it visible now, ma'am?
No, ma'am. Only a file explorer window is open.
Actually, I have opened it. I don't know how to.
You will have an upside arrow where you have option of sharing your screen.
Yeah, it shows me your shares. You are screen sharing.
Now click on those slides, ma'am.
Sorry?
Click on your presentation. Click on your PPT presentation. Just try to open that.
Is it okay? Can you see this?
No,
Showing
no.
it's sharing.
Can you see the slides, Ms. Donia?
Sorry?
Are you able to see the slides?
Yes, I can see.
Okay, so you can just go back to the Zoom window and now you again try to share screen.
Okay.
And then the options that is shown, select the one with the slides. You can just come back to the Zoom meeting and then select screen share.
I'll select share again. Is it visible now?
Yes, it's visible. Yeah.
Can I start now?
Oh, yes, please.
Okay. Good afternoon. First of all, I'm thanking the organizing team for allowing me to present this. My topic is association of autonomic imbalance with the duration of disease in patients with chronic lung diseases. Sorry. I'm not able to move the slide.
Can you try putting it in full screen?
Just make it a slideshow, ma'am.
I'm not able to see the slideshow option.
There is a play button in orange in the bottommost
Yes.
toolbar. Yeah.
Yeah, it's fine now. Okay, thank you, ma'am. And my study is actually on the chronic lung disease group. Actually, it is a little different from the microbiology topic, and I'm thanking you for that. For
Still, it's
allowing
not
me to present
slideshow.
this. Am I audible clearly, ma'am?
Yeah, you're audible. It's still not in slideshow.
Okay. And the topic is autonomic imbalance in chronic lung diseases with the duration of their disease. And for that, I have selected the COPD and chronic obstructive lung disease patients as well as interstitial lung diseases. Their autonomic imbalance I have been checking with the heart rate variability, HRV, which is an invasive tool for assessing the autonomic functions, and that is assessed in both the study group, which are the chronic lung diseases, and I have seen whether there is any autonomic imbalance in them, and if it is there, then how it is affecting or how it is associated with the duration of their disease. And the duration of the disease of these group were like starting from 2 years to 7 years. That was the disease duration of these groups, and I have analyzed whether these duration of the disease is affecting with the sympathetic or parasympathetic, or the autonomic functions are affected or not. The aim is to study the association of autonomic imbalance with the duration of the disease in chronic lung disease. And in objective, I have divided this chronic lung diseases into obstructive and restrictive lung diseases. Obstructive will be the chronic obstructive pulmonary disease, COPD. And in restrictive lung disease, they are mainly focused on the interstitial lung diseases.
Please change the slide. You're on the still first slide, and it's still not.
I'm changing the slide, ma'am.
No, I'm not.
Actually, I'm in the materials and methods. Am I
I'm
still
seeing
in
your
the
first in your title slide only.
One second, ma'am. For me, it is moving. Sorry. Can you see the change, ma'am? Is it changing now?
Yes, yes. Yes,
Okay.
I can see now. Yeah.
Okay, thank you, ma'am. Sorry for the confusion. And this is the aim. In aim, I have it is to study the association of autonomic imbalance with the duration of disease in chronic lung diseases. And in objective, I have divided these chronic lung diseases into obstructive and restrictive group. The obstructive group will be the COPD patients, and restrictive group is the interstitial lung diseases. And in materials, I have selected the chronic lung disease. I have divided into COPD and ILD cases. A total of 52 COPD patients and 43 ILD patients were there, which were coming under an age group of 40 to 60 years. And they were included in the study, those who were able to perform the PFT, pulmonary function test. And in the heart rate variability, I have selected the parameters like low frequency and high frequency heart rate variability ratio, and the normalized units of the low frequency and high frequency bands of the heart rate variability. And in PFT parameters, I have selected mainly the FEV1 percentage predicted, FEV1 FEC ratio, FEV1 ratio, then peak expiratory flow rate. These are the values I have, parameters I have taken for the pulmonary function test. And the inclusion criteria was the COPD and ILD patients who are able to perform PFT. Because heart rate variability, there was nothing much to do for as such. And in PFT, they should perform that particular. They have to breathe through that instrument. Therefore, some were not able to do that in those who are in severe conditions. So I have selected only those who can. Those who were able to perform the pulmonary function test. And exclusion criteria was like those who are having any autonomic function, the diseases, any diseases which were affecting the autonomic functions like nerve degenerative disorders, conjunctive heart failures, tuberculosis, conduction disorders, all those kind of diseases, if any of them were having, they were excluded. And in the results, I have found that there was a significant relation between the heart rate variability and pulmonary function test in both the groups. And all these tests were done by the student t-test. And some data I have correlated, which was done by Pearson correlation coefficient. And in this table 1, I have checked whether the COPD, whether the heart rate variability is having any difference in COPD and ILD groups, and it was showing some significant difference in both the groups with this heart rate variability, like the parameters like low frequency and high frequency band, and low frequency band which is normalized unit, and high frequency band with normalized unit, and especially the values were more significant with the LF normalized unit. That means in those who were showing
Those who
more,
have
sorry.
Please go ahead.
In the LFNU means that means the patients are having sympathetic predominance, and HF value means there will be parasympathetic predominance, and so I got values like more predominance with the sympathetic. That means it was showing some difference in the autonomic function. And in this table 2, I have checked the correlation between the heart rate variability in PFT parameters in the COPD patients. And in this, I couldn't find much difference. This correlation among these two parameters in the COPD patients were not showing any kind of significance, but their heart rate variability in both the cases, both the study groups were showing significance. And also I have correlated with the duration of the disease. That was the major point I have done here. According to the duration, I mean, as the disease duration is increasing, there were significant difference in these values, especially in the heart rate variability, but not exactly with the pulmonary function test. I couldn't find much difference with the disease duration, but I could find a difference in the duration of disease with the heart rate variability. And in this table, it is clear that the heart rate variability with the duration of the disease, as the duration increases, there was significant change, but there was not a significant change is observed in pulmonary function test with the duration of the disease. This is done in both the COPD and ILD cases. In both, it was showing like that. Heart rate variability was showing highly significant values with the duration of the disease. Like the duration of the disease, as I told in the beginning, that the duration was from 2 years to 7 years. So as the duration increases, the significance was more with the heart rate variability, but not with the pulmonary function test. So in summary, I could find that the duration of disease is affecting with the heart rate variability, which is an indicator of the autonomic function. That means in them, in this chronic lung disease patients, actually the autonomic dysfunction is seen, and that is more prominent with the when the duration of the disease is increasing, even with the medication. But maybe with the medication, the PFT values are fine, or they don't have any significance, but heart rate variability was not highly observed among all these groups. So it is showing that it is having a difference. So we have to consider the heart rate variability, or the heart functions or cardiovascular functions should also be taken care along with the pulmonary functions or the medications along with the pulmonary function. And in both the study groups, I could find highly significant values.
Ma'am, I'm sorry to interrupt you. The time is running out, ma'am. So kindly wrap it in 1 minute.
Okay, ma'am. Thank you. In conclusion, it is proving that there is an association between the chronic lung disease and autonomic imbalance with the duration of the disease, and this is what my study is. And these are my references, and I acknowledge my guide, Dr. Gino Merlin Koshi and Dr. Ajit, who had supported me in this study. Thank you. Thank you,
Thank you, Ms. Tonia. I have few questions for you.
Okay.
Did you follow these patients from 2 years, as your duration is 2 to 7 years? So you saw the case when they were diagnosed and you followed those patients for another 4 years or 5 years?
No, ma'am. I haven't followed them, but I took their history and I checked with the values.
Okay. Another question. These patients will be on steroid, either oral or inhaled. Do you think steroids are affecting their autonomic nervous system, or drugs are affecting?
That is the one drawback of this, because I haven't noted that way. But the steroids, maybe because of the steroids, they don't have any pulmonary disorders. I mean, increase in the pulmonary function disorders, but no particular drugs were given for the cardiovascular function. Therefore, maybe because of that, it was showing increased heart rate variability, but not significant values with the pulmonary function disorders.
Yes, so that should be limitation of your study. Thank you. Thank you.
Thank you, ma'am.
Thank you, ma'am. For the next presentation, I invite Dr. Sadaf Amreen from Sharda University. Dr. Sadaf Amreen. If she's not available, Dr. Sandhya, are you available?
Yes, ma'am. Yes, ma'am, I'm here.
I'm Dr. Sadaf.
Oh,
I'm
okay. You're on Dr. Shrutika's
Yes, I'm on
Okay.
Dr. Shrutika's laptop.
Okay, sure. Please go ahead.
Good afternoon, everyone. I'm here to present a case series, a hard hit of new epidemic disease mucormycosis in Indian population during COVID-19 pandemic.
I'm not able to see your screen, Dr. Shada.
Okay, ma'am. Can you see
Yes, I can see now.
Okay, ma'am. Okay. So here I am presenting a case series, a new epidemic mucormycosis in Indian population during COVID-19. Myself, Dr. Sadaf Ambreen, I am Associate Professor in Sharda University. The introduction, the year 2020 was hit by entry of new disease COVID-19, which was declared as a global pandemic by World Health Organization in March 2020. The pandemic continues to be an ongoing public health concern with more than 162 million cases recorded and more than 3 million deaths globally. With the escalator of cases of this disease worldwide, a numerous potential complications from COVID-19 are being increasingly appreciated, including the heightened vulnerability of the secondary bacterial and fungal infection. Since the start of this COVID-19 pandemic, there has been renewed interest about secondary infection. During COVID-19 situation, India saw a surge of another rare fungal infection, that is mucormycosis. So mucormycosis, a fungal infection commonly affecting structure within head and neck region like ear, sinuses, orbits, and later on in brain. Mucormycosis is known to affect immunocompromised patients, especially those with diabetes mellitus, prolonged corticosteroid use, and that solid organ transplant recipient, and neutropenic conditions. So the country recorded almost 28,252 cases of mucormycosis from 28 states till June 2021. Data which are collected showing 24,370 cases are cases with history of COVID-19, whereas 17,601 cases with the history of diabetes. So aim of objective of my study or cases here, we share our clinical experience of the sum of mucormycosis cases that was mostly in maxillary antrum with uncontrolled diabetic mellitus managed at our college. The cases reported are with the following aims and objective. First, to emphasize the need of early diagnosis and prompt aggressive surgical management followed by the institution specific antifungal chemotherapy to ensure a favorable outcome. Second, to draw attention toward the histopathological presentation of mucormycosis. And third, to create awareness that have high suspicion of mucormycosis cases in immunocompromised patients. Case series, first case series, we describe our clinical experience with series of cases which are reported during COVID-19 pandemic in 2021. Since there was a sudden surge of mucormycosis and this was also an infrequent disease, so there was a diagnostic as well as therapeutic dilemma for those who aren't conversant in its clinical presentation. It was noted that reported cases of mucormycosis was associated with uncontrolled diabetic mellitus managed at our hospital. So case 1, a 42-year-old male patient reported at Sharda Hospital, which was referred to Sharda School of Dental Sciences, with swelling and tenderness in right maxillary region along with mild fever from past 20 days. Pain were associated with the swelling region of face without involving orbital part. Patient have history of diabetes mellitus from past 8 years with medication. Patient was fine 20 days before. He developed mild fever during already occurring COVID-19 situation. He had undergone with some local consultant doctor for RT-PCR test, which was negative. He was prescribed with some medication along with steroids for 7 days. Due to medication, fever subsided, but patient started complaining of pain in upper right region. Pain was acute in onset, gradually progressive, initiated from upper jaw region. Palpation showed tenderness over entire palatal region. Patient complained about loosening of all the teeth. During intraoral examination, discolored palatal mucosa was noted with multiple draining abscesses in maxillary region. Radiographic investigation was done and paranasal sinus view was advised, and which shows haziness of maxillary sinus with erosion of lateral sinus wall. A CT scan of face showing heterogeneous soft tissue density involving bilateral maxillary alveolar bone process and palate. These are the CT scans which are showing bilateral palatal swelling. Now, biochemical investigation. Biochemical investigation revealed an elevated fasting blood sugar level of 230 milligram per deciliter and elevated postprandial blood sugar level of 356 milligram per deciliter. A provisional diagnosis of maxillary osteomyelitis was made. Biopsy of hard tissue along with soft tissue, adjacent soft tissue was excised under local anesthesia and sent for microbial and histopathological examination. Microbial examination revealed broad aseptate ribbon-like hyphae, and this was later confirmed by culture. Microorganism examination revealed broad aseptate ribbon-like hyphae, and this was later confirmed by culture. History of mucorales revealed finding like non-pigmented, a wide 5 to 20 micrometer thin-walled ribbon-like hyphae with pauciseptation with the right angle branching. On histological and eosin staining under 40x magnification, hyphae will find empty looking. So result and discussion, histopathological examination shows with hematoxylin and eosin stain readily identify non-septate mucomytic hyphae. Following confirmation of diagnosis with 4 to 5 days, the patient was hospitalized and his blood sugar level was controlled by the physician. On surgical exploration, the necrotic bone piece was found to be loose and was removed, leading to opening of the antrum. On examination, the antrum lining was found to be thick and fibrous. Maxillary sequestromy was done, and total antral curettage and complete debridement was carried out. And the wound was secondary closed using an iodoform pack. Incisional biopsy was taken from the maxillary alveolar ridge. The patient was administered a single daily dose of liposomal amphotericin B1 milligram, 1 milligram per kg body weight, and an infusion in 100 ml of 5% dextrose over 1 to 2 hours for a period of 15 days. The blood urea and creatinine level was monitored for nephrotoxicity. The patient was sent home on 4th day with advice to take injection and to come for regular follow-up. 2 weeks later, signs of healthy granulation tissue was seen and the wound healed and eventually in around 3 months. These are the histopathological pictures. These are multiple draining abscesses noted. If you can see these on maxillary area. And these are sequestrum bone which is removed surgically. So case 2, 65 size with complaining of pain and swelling in upper teeth region for past 1 year. The patient had medical history again, diabetic and hypertensive. She was undergone medication for past 2 years. Along with that, patient give history of hospital admission 1.5 months back because of weakness and high fever. Patient was also on a central line oxygen for 30 days. RT-PCR test showed negative report. She was apparently well 1 month back until she developed a pain on both sides of the face, which was acute in onset. Pain was associated with swelling over the both sides of the face, not involving the orbital region. Patient also complains of mobility of the teeth, which was insidious in onset. On a local examination, a single solitary lesion of size approximately 2 into 1 centimeter was present in relation to right maxillary posterior region. On palpation, tender was present over the entire palatal region. Intraoral examination showed necrosis as well as exposure of the buccal cortical bone in relation to right maxillary posterior region with upper overlying inflamed palatal mucosa.
Ma'am,
Radiographic.
I'm sorry to interrupt you, but the time is running down, ma'am. So can you please jump to the conclusion? There is just one more minute.
Yes, I'm okay. So again, radiographic picture was taken, and these are the summary of these 2 cases. Amphotericin as an antifungal drug given. Result is mucormycosis is being commonly among COVID-19 patients, especially thanks to physiological stresses like high blood heat, osmolarity, high hypoxia, which are common condition when affected with SARS-CoV-2. Also, these patients undergo heavy intake of steroid using oxygen mask and ventilator to combat SARS-CoV-2 infection, which turn as an entry pass to the body for the mucorales fungus. Further, these mycosis could impact the COVID-19 in 2 ways scenario. when the COVID-19 patient who have diabetic as comorbidity create a acidic environment that permit a singular environment for these fungal to grow. And also thanks to hypoglycemic condition, there is a decreased production of T cells and immunosuppression leading to a cytokine storm. Heavy intake of steroid also lead to the same condition. So early imaging, laboratory investigation along with mucormycosis with combination of the all ENT surgeon, maxillofacial surgeon, ophthalmologist, they can use, we can combinedly, we can treat this mucormycosis. Aggressive clearance of pathology is required. Maxillofacial surgeons are also required for removal of the sequestrum bones. So result is showing medication. First line of antifungal is amphotericin B therapy, and second line will be injections and pesoconazole tablets. So conclusion is use of steroids which act as the main line of therapy for COVID-19 infection to reduce mortality, act as an exacerbation for factor to suppress the immune suppression response. Diabetic mellitus is another important risk for factor, and the presence of both having additional effect in causing secondary fungal infection. COVID-19 pandemic emerged with mucormycosis, a rare infection, as epidemic in Indian population in 2021. Early diagnosis with radiological as well as pathological investigation followed by surgical debridement and antifungal treatment had led to the to speedy recovery. Thank you.
Thank you, Dr. Shada.
Thank you, ma'am.
So your study was 2 years back, right? Your study was done in 2021 when there was pandemic. What is the scenario now in your patient? The patient who are on steroid because of some other condition, are they presenting with mucormycosis?
Ma'am, the condition is still going on. The treatment is still going on because it's a very aggressive procedure. And those who are suffered from COVID-19, they are still coming with these fungal infection. But the percentage or data is less as compared to those condition in epidemic condition of COVID-19 in 2021.
Okay, so how long it takes? Did you ask for QH apart from your histopath and other examination?
Pardon me,
Microbiological.
ma'am, I am not
Did
able
you
to
ask
hear.
for microbiological investigation along with your histopathological diagnosis?
Ma'am, actually at that time it was emergency cases, so we just took the pathological condition and we confirm on the basis of radiology and clinical pictures, and on the basis of that we have treated these cases, and these mucormycosis cases are still going on. We have given the
Yeah, I
microbial
have one more question. Your microbiological investigation gives you the diagnosis in few hours. Radiology gives you in a few hours, but histopathology takes days to confirm it. It doesn't happen in a day. So if you add microbiological investigation along with what you are doing, that will add you benefit for your study. And never say anything thanks which is detrimental. No thanks to COVID-19, no thanks to steroid. Is a factor. COVID-19 is a factor. Steroid is a factor.
Yes, ma'am. Yes, ma'am.
Yeah. Thank you. Thank you.
Thank you.
Thank you very much. Before inviting the next speaker, I would just like to kindly request the participants to stick to the time. While we completely respect and appreciate the time that you've taken to make the slides and the presentation, I completely respect that. But also keeping in mind and respecting everybody else's schedule and time, just a kind request. And I will also remind you probably 2 minutes ahead to help you pace the presentation. So on that note, I want to invite Dr. Sandhya Manjri for the
Yes,
presentation.
ma'am. I'm here. Yeah.
Yeah, please go ahead. You can
So
see
can
a
you
slide.
able to see my screen?
Yeah, I
ma'am?
can. Yeah, we can see a slide. We can hear you.
Yeah. Okay. Myself, Dr. Sandhya Tambeka Vanzari. I am from the Dotebandhu Science College, Gondia, Maharashtra. So here I am presenting a paper on the generic versus ethical drugs against neuropathogenic Escherichia species. So the presence of the microbial pathogens of the microbial pathogen in the urinary tract with the associated symptoms in the UTI infection. Second most prevalent bacterial infection after respiratory tract infections in the women. So UTI are the mostly common among the female population. Hence, about the 1% of the school-age girls, women through the childbearing years are infected with the UTIs. So, as the incidence of infection in the females increases directly with the sexual activity and the conditions, about the 25 to 30% of the women between the ages 20 to 40 years come across with the UTI infections. The Escherichia coli is the most common cause of the uncomplicated community acquired UTIs in the both inpatient settings. So the knowledge of the main bacteria usually involved in the UTIs and their antimicrobial susceptibility necessary for the appropriate empirical therapy and prevention of the emergence of the antibiotic sensitivity. So a generic drug is a drug that is not branded, but it is similar to branded to the reference and ethical drugs terms of the doses, administration and performance. It has some composition and the biological composition is equivalent to the brand drugs, but it is not typically sold at the substantial low rates as compared to the branded drugs. So therefore, an attempt was made to find out the efficiency of the generic and ethical drugs against the neuropathogenic Escherichia coli, that is especially Escherichia species. This will enable the users and medical practitioners to select the effective and affordable drugs for the treatment of UTI infection due to the neuropathogenic Escherichia coli. So material methods of we are collecting total 50 urine samples were collected from the pre-autoclaved sterile bottles in the sterile bottles containers from the 5 different pathology labs in the government hospital and process immediately after arriving the laboratory. So the collected 1 ml of urine sample were inoculated in McConkey agar medium with the help of the sterile spreader and incubated at the 24 hours for the 37 degrees centigrade in an incubator. And after incubation period, pink colored colonies were picked for the maintaining the maintaining the further process. And further, their analysis with the cultural microbiology and biochemical tests were performed to identify the microorganisms isolated from the collected urine samples. So here are the total that is 8 antibiotics are used, generic and ethical antibiotics are used, amoxicillin, potassium, ciprofloxacin, azithromycin, aflatoxin, levofloxacins, amoxicillins, and ciprofloxacins and ciprofloxacins. So that are the antibiotics. So this is the picture which involves the generic drugs and ethical drugs with the different types of the 8 types of the antibiotics which are used against the Escherichia species. So ethical and generic antibiotics were purchased from the local medicine market, and the 200 milligram tablets of each antibiotic were dissolved in the 10 ml of the sterile distilled water and stored it for the further uses. After autoclaving and cooling at a room temperature, nutrient broth was inoculated with the Escherichia species from the isolated from the urine sample, and tubes were incubated. After solidification of the nutrient agar, plates were lawn with the 1 ml of the broth cultures, and total 8 wells was made in the sterile borer with the help of the sterile borer, and were added to the 50 microliter of different antibiotic solutions. Separate plates and same isolates were used for the loading generic and ethical antibiotic solutions. Plates incubated for the bacteria for the certain types of the growth for the 24 to 48 hours, and incubated plates were observed for the clear zone of inhibition due to the loaded antibiotics, and then observed for the diameter, which are zone of inhibition which is caused by in the diameter. So our total 50 samples were collected from the 5 pathology labs located in the Gondia district. So 44 Escherichia species were isolated from the urine samples, and identification isolates were done by the morphological and cultural characteristics. So these are isolates are the gram negative, short rods, arranged in a single of pairs. It is pink color, smooth circular rays colonies in the McConkey agar medium, and it is a biochemical test is positive for the certain types of the identification. So here's the tables which are use the comparative analysis with the ethical and generic drugs after 24 incubation. So here we can see that the total about 19 samples of the Escherichia from which is collected at 43%, 43.2% show the sensitivity. Cifeflexin, that is a 95%, that is more sensitive, and the erythromycins, that is 93%, and the aflatoxin, that is 95%. Ciprofloxacin and levofloxacin shows the most, that is 100% sensitive to the particular Escherichia species by the ethical drugs. And the generic drugs in the generic drugs, the levofloxacin shows the 100% sensitivity, and the aflatoxin shows the 100% sensitivity. So this is about the particular 24 hours incubations. So the 100% species isolate from the urine sample collected from the different pathogens sensitive to both ethical and generic levofloxacin drugs, whereas the percentage of the isolates from the is sensitive to both ethical and heterogeneous, that is amoxicillin trihydride after 24 hours incubations. Then we again they're further incubated for the 48 hours, and then we deals with the percentage of the sensitivity for the particular ethical and generic drugs. So we can find out that cefixlexin, that is 100% sensitive and same as that of the generic drugs. Then again erythromycin, that is 93% is ethical drugs and generic drugs is 100% sensitive. Then aflatoxins, that shows the zone of inhibition in both ethical and generic drugs. And then ciprofloxacins, that shows the ethical drugs is more and the generic is the less. And levofloxacin are 100% sensitive to that particular retarded to resistant to the Escherichia coli.
Just 2 more minutes, ma'am.
Yes. So this is the particular slide. The slide is shows the zone of inhibition which is created by the formed by the particular antibodies, that is ethical and generic drugs. So conclusion recommendation: there is no significant difference in the sensitivity of the uropathogenic Escherichia species against the ethical and generic drugs. Hence, it is suggested to use generic drugs instead of the ethical drugs, because generic is more economical than the ethical drugs, and this reduces the burden of the high cost of ethical drugs on the particular patients, which lowers the expenses on the medicines. So medical practitioners should benefit from this study, as it is information that about the potent and anti-uropathogenic Escherichia drugs. So hence, by prescribing these particular drugs, they can avoid the chances of prolonged recovery of the UTI patients. So it is more economical than the ethical drugs, and because they have the same near about the same activity or antibiotic activity on the uropathogens, which is found by the urine sample. So here all about my presentation. Thank you very much.
Dr. Sandhya, please go to your result and conclusion slide.
Okay. Yes, ma'am.
What do you understand by ethical drugs?
Madam, this is the more complex compositions which are involved, and there is a expensive drugs which are not capable of by the patients, but generic drugs is more.
You mean to say second generation or higher generation of drugs, right?
Yes, ma'am, somewhat. Yeah.
Okay.
Yeah.
So you did study in your patients? How many patients you took?
Ma'am, I'm not going through the patients. I'm going through the only 50 samples from the pathogenic labs. Sorry, patho labs.
So you're taking the samples from lab, you're putting the cultures?
Yes, ma'am.
And checking the sensitivity?
Yes, ma'am. We are not directly contact with the patients.
Okay. Yeah. So what about your ethical clearance and consent form?
Sorry, ma'am.
What about did you get the ethical clearance from your institute for this study?
No, ma'am. No, ma'am. This is the dissertation study, MSc dissertation study for the PG students, ma'am.
So you must have got clearance from your ethics department before you started your study. Or do you have do you get patient consent? Or
no, ma'am. No, ma'am. It is directly from the pathology lab, so I am not taking any consent from the patients.
So consent form is waived off, right? Am I considering the right? But you must have some ethical clearance.
Okay.
Any human specimen which you test, whether you take patient consent or you waive off the consent, should have ethical clearance for your ethics committee.
Okay, ma'am. Yeah.
Oh, thank you, Dr. Sandhya.
Yeah, ma'am. Thank you.
Thank you, Dr. Sandhya.
Yes, ma'am. Thank you.
For the next presentation, I invite Dr. Shani T. John.
Sorry for disturbance, ma'am. Ma'am, this 1:20 p.m. is my slot.
I understand, sir. We are going according to the agenda. We have been delayed by like 10 minutes or something. So instead of Sujata ma'am, Dr. T. Shani John will present. After that will be your slot, sir.
Then, ma'am, I can go for my tutorial for MBBS students for 20 minutes.
So you will be coming back in another 20 minutes?
Yeah, ma'am. My class is running. A practical session is running.
So you can finish and join us.
Yeah, okay, ma'am.
I will shift your slot at the end.
Thank you, ma'am. Thank you.
Yeah, ma'am. Audible?
Yes, ma'am. And the slide
Okay, my
is visible
screen is
too.
visible?
Yes, ma'am.
Okay, Shani, Assistant Professor from Center for Marine Science and Technology, MS University. I'm going to present a topic on antimicrobial properties of sea urchin Diadema cytosum collected in Kadiyapatnam coast, Kanyakumari against human and fungal pathogens. So actually, as we are going on with multi-drug resistance by both gram-positive and gram-negative.
Please put the slideshow.
Yes, ma'am.
Dr. Shani, please put the
Okay, ma'am.
slideshow.
Okay. Okay, I think I have done with the slideshow.
It's not reflecting for us, ma'am. It's still not in slideshow.
So I have done with that, but
okay, go ahead.
Okay, so as we are going on with multi-drug resistant by different types of microorganism, it is a prime aim to go on with a search for certain different kinds of antibiotics that may not interfere with the resistance of the bacteria. In such cases, the oceans or the floors which provide us with a large amount of active compounds from different sources, like even the sediment sample can provide us with different types of organisms, and also the marine organisms which are dwelling in the environment can also provide with certain kinds of compounds. In such instance, I have gone with the collection of sea urchin from the Kadiyapatnam coast, which is available in our nearby area. I have collected this sample with the help of the local fishermen, as well as I have also seek the help of scuba divers. I have collected the sample, and I have gone for certain objectives like to determine the antibacterial activity, antifungal activity of the extracts, especially the gonad and the spines which are collected from the sea urchin, and also to isolate the pathogens, associated pathogens from the sea urchin, and also to analyze its antagonistic activity with the pathogens. Next, the crude extracts, that is the gonadal and the spine extracts, which showed best activities were analyzed for minimal inhibitory analysis, and it also is being compared with the commercial antibiotics to characterize that particular compound. And later on, I have characterized that particular compound using UV and FTIR to see whether the presence of a particular element. So my materials is like, I will have collected the samples from Kadiyapatnam coast. The samples were collected in aseptic conditions, washed several times with the seawater to remove the debris, and it is carried out to the laboratory in a polyethylene bags. Then again, after coming to the lab, we have rinsed the sample in fresh water to remove the saltwater contaminants and to get rid of the remaining impurities which are present in the epiphytes, that is the organisms.
They were identified with the help of the taxonomist and also with the data which is available in the internet. And this is the area from where we have
Please change
collected
to your slide. Please keep changing your slide when you're moving ahead.
Yes, I'm changing, ma'am. I think I have some problem with my internet connection.
No, I think your internet is fine. Introduction, I can see only introduction slide. Just try to click or you rejoin as ma'am said, come and rejoin through Zoom.
Okay, I have stopped sharing. I will try to share it now. Is it visible now? Am I audible?
Yes, ma'am, it's you're audible. The slide is not in slideshow, but we can see the introduction slide.
Now, can you see my materials method section?
No, only introduction is seen, ma'am.
Shall I rejoin?
Okay, I think so. In the meantime, if Dr. Mrinalini is free, we can go ahead with your presentation, ma'am. Dr. Mrinalini, are you there?
Yes.
Can we have your presentation, ma'am? Is it ready?
Yes, it is ready.
Okay, yes, ma'am. Please go ahead.
है ना वो प्लानिंग का
Okay, just a sec. Is my screen visible?
Not yet, ma'am. It says, yeah, it's visible now. The slides are visible.
Okay. We have conducted a small survey on awareness about sexually transmitted infections among young individuals of science and non-science background. Basically, the aim was to gather an information about whether there is an awareness about these STIs amongst young individuals, whether of any background, but in the age group of 18 to 30. So there are various diseases such as syphilis, gonorrhea, chlamydia, trichomoniasis, which are caused by this in bacteria, viruses, and parasites, including HIV, of course, and herpes simplex virus. But as there is less awareness, there is a risk of this contracting in young individuals, especially when people go for higher education or for their career outside their hometowns. They come in contact with the people of other socio-economic background, and there could be such mishaps which may lead to the sexual contact followed by the sexually transmitted infections. So we know that there are common ways which are generally for transmission of such diseases, including sexual contact, including vaginal, anal, and oral sex. Then during pregnancy, it will be transferred, can be transferred from mother to child. Kissing at times can include or can transmit such diseases, especially in case of herpes simplex virus. So in 2020, there was an estimate of about the new infections, which WHO had estimated 374 new infection cases were arising, out of which in 2020, they found that out of which 1 of 4 infections were STIs, sexually transmitted infections. So that was really alarming. In 2021, they also announced Awareness Week. WHO also announced Awareness Week for this sexually transmitted infections. Now, the aim was to assess the knowledge, attitude, and preventive practices which these young individuals are following, whether they are following or not, or whether they are even aware of it or not. Now, to assess this awareness about sexually transmitted infections, the objectives were to assess the awareness about sexually transmitted infections amongst young adults. What is their attitude towards these infections, whether they are aware of any preventive measures, whether amongst science and non-science background students or young individuals, not necessarily students, whether there is any difference or not. Okay, and then through survey, creating such awareness. That was this was done by one of the MSc students. So there were 167 responses which we could gather in the age group of 18 to 30 years, and basically in the area Mumbai. And this was online because, as you know, this was the COVID period. And so the survey was conducted online by circulating the questionnaire through Google Forms. It had this data collection was done in 3 sections: the demographic details such as age, profession, and education background, the knowledge level in section 2, and section 2 and section 3 were pertaining to the sexually transmitted infections particularly. So whether they have heard about it, what was their source of information, whether they are aware of routes of transmissions, about the symptoms, about the complications, whether these young adults are aware of it or not. Then whether what is their attitude towards it, what practices, preventive practices should be followed, whether they are aware of it. That was asked in the questionnaire. To give you the gist of results, what we obtained, most of them had heard about sexually transmitted infections. But as we can see, more of females have heard about it. The ratio was slightly more towards female. So female had heard about this sexually transmitted infection, which was a good thing. Then those in the age group of 22 to 25 were more aware of it rather than in the 18 to 21 years age group. Then when they were asked about which of these are they know about sexually transmitted infections are, so surprisingly trichomoniasis caused by the parasite, people were aware of it. Okay, sorry, people were not aware of it. Students were, these young individuals were not much aware of it. Then even chlamydial infection, there was not much awareness, but AIDS or HIV infections were known. Then when we tried segregating the data as being science and non-science, gonorrhea and syphilis were more known to people from science background, also HIV, as there was a slight difference with respect to HIV. But yes, at least with respect to gonorrhea and syphilis, more awareness, even genital herpes was, students were aware of genitis. Just a second, I was trying to put the...
Ma'am, there's 2 more minutes.
Yeah, sure. In case of routes of transmission, there was not much difference with respect to routes, awareness about routes of transmission, whether it is through sexual intercourse, blood transmission, or all. Basically, all of these are included in the transmission of the disease, kissing to the lesser extent than sexual intercourse and blood transfusion. But even there was not much difference between science and non-science young individuals about the awareness. Then about the symptoms, ulcers in genital organ, most of the science students were aware, even about the discharge from vagina, science students were aware, more aware of that. So here, science students were more about of the symptoms of the sexually transmitted infections. Then regarding the complications, again, there was not much difference, and both of them were not aware about the complications such as stillbirth or genital cancer or ectopic pregnancy. Young individuals, overall, the awareness was less. Although 50%, we can say 56% of the science people were knowing, so they said all of the above, but still 50% almost were not aware of this complications. Then about the preventive measures or the attitude, about the attitude, whether condoms should be used, whether use of condoms is advised or not. Yes, most of them said use of condoms should be there, but still some of them were not sure about the use of condoms, whether it will prevent the disease or not. And regarding the preventive measures taken, the attitude or rather whether the screening is necessary for this for the sexually transmitted infections. 50% of them agreed with science background as well as in non-science background. 50% of them agreed to the fact that yes, screening should be done, and whether if the symptoms are seen in the partner, whether the individual should go and seek the treatment. That again, yes, most of them were agreed to. Then about the vaccine, about the human papillomaviral vaccine, many had not heard about it. So that was again little alarming. Although science, yes, science, most of them had heard, 86%, but non-science, hardly anybody was knowing about it. And when it was asked, will you abstain from the sex? Most of the science people, yes, both of them rather said that yes, it will be. Oh, sorry, they denied that. No, they said no, no. So they were not aware that abstaining from sex will also help them, give them some time for the recovery of the treatment. So the knowledge about this, whether abstaining from sex and whether mutual monogamy should be preferred, they were not very clear. Both of them were not very clear. And use of condom, however, most of them were agreeing to it that it would curtail the infection. About the vaccine, science background people were knowing more about it than the non-science background people about HPV, human papillomavirus vaccine. So overall, the impression was that there is no significant difference between between science and non-science background students or individuals, young individuals, about the sexually transmitted infections. So more efforts are needed in this direction so as to prevent it. This is one of the Sustainable Development Goals, 3, part of SDG 3, wherein health regarding reproductive organs has been advocated. So there more such awareness campaigns are needed, and even young, both ways, government as well as the young individual should have more knowledge about it, and they should avoid the unprotected sex. These are some of the references which we had referred to. Thank you.
Thank you, Dr. Mrinalini.
Yes.
I have few questions for you, Dr. Mrinalini.
Yes.
How did how did you confirm it was online, right? So
yes, it was.
Are known people or random people selected?
Some of them were known. Some of them were known, but
okay.
not
so
all.
How
were
you
known.
confirm their age before sending them questionnaire, either through message or mail? How did you confirm that they are 18 to 30 years?
For the age? Okay,
yes.
No. Okay. No, there was no such. Sorry, we did not think about that.
Okay.
I missed that.
And if you could have projected the empty questionnaire at the beginning of your slide, that could have been one
sure.
good point. And did you, once when you saw that HIV and use of condom, these are normal things which I think every individual who is going to adolescence should know. When you came to know that these many this considered to be higher percentages who doesn't know the use or harmful effect of these, did you send them the feedback to aware them?
No, we haven't done in that
Please
direction.
do it.
That's a good
That might
suggestion.
be.
Yes, that's a good suggestion. But yes, in our college, we did to some extent creating an awareness through our students, microbiology students, but not to the outsiders. We couldn't reach. Yes.
So you can, who all have given the, like they don't know, you can create an online template or awareness, some online model and send to them. Thank you. Thank you so much.
Thank you. Thank you.
Thank you, ma'am. If Dr. Shani Tijon is ready. Yes,
ma'am.
Can you share your screen, ma'am?
The screen is visible now?
Not yet. It's visible now.
Okay. So thank you for the chance again. Myself, Shani. I'm going to present my topic on antimicrobial activity.
Can you please continue from where you left, ma'am, instead of from the beginning? Because that'll be only about 5
Okay,
minutes for your
okay.
presentation.
Then okay. So like so we'll go on with materials and methods. I have collected the sample from Kadiyapattanam coast near our coastal area and brought it, washed it thoroughly in the seawater. Then the crude extract, that is after separation of their shell, the gonad and spine samples were separated and it was crushed with the methanol and other solvents to extract the particular crude extract. Then again, we have done with antimicrobial assay with the crude extract using the method of Nathan et al. 1978, that is agar-well diffusion methods. Later on, the crude extract, the gonadal and the spine extract, which showed best activities, were analyzed for minimal inhibitory test by following the method of Andrews. Then the associated microorganisms with the sea urchins were also analyzed for the antibacterial activity against different human pathogens. And next, the gonadal as well as the spine extract, which showed best results, were compared with the commercially available antibiotics. Finally, the extract were also characterized using UV FTIR analysis. Results and discussions. The sea urchin collected from the current study was identified as Diadnama cytosum with the help of the taxonomist as well as with the help of the internet. This species then were screened for antimicrobial activity. The extract, both the gonadal as well as the spine extract, were good enough to carry out inhibitions of both the fungal pathogens as well as the bacterial pathogens. That too, some of the, that is the gonadal extract was good enough to inhibit the growth of human pathogens rather than fungal pathogens. It showed inhibition zones of greater as well as less than 10 mm diameter. You could see from the results that the methanol, chloroform, I have used different solvents for the extraction of gonadal as well as the spine samples from the sea urchin, where the methanol as well as the chloroform extract showed some amount of inhibition against the human pathogen, where the methanol extract can inhibit Vibrio cholerae by 9.5 mm, whereas that is chloroform extract can inhibit E. coli by 8 mm, followed by several other pathogens. And the antifungal activity of the particular extracts like gonadal as well as the spine samples were also analyzed against certain fungal pathogens which were procured from Agricultural University, Coimbatore. There too, the gonadal samples were good enough to inhibit Candida albicans and Rhizomycor mehi with inhibition zones of around 5.5 and 5 mm. Then the extracts which showed best activity were column fractioned
to analyze, to extract the particular compounds which were which can inhibit the pathogens. Column fractions were done with the help of ethanol methanol. That is the 2:2 ethanol methanol fractions were good enough to carry out or elute particular compounds. This method was followed using the procedure of Wright 1908. The methanol phase was then fractioned using normal phase silica gel, column chromatography with the step gradient solvent system. This is the thin layer chromatogram we have done to prioritize the compounds which can be eluted from the column fractions. Then the fractions which were eluted from the columns were also analyzed for its minimal inhibitory concentration against the test pathogens, both against the human as well as the fungal pathogens. There too, this 25 microgram concentration of the gonadal extract was good enough to carry out the sidal effect of Pseudomonas aeruginosa and also against the fungal pathogens Candida albicans. And whereas 50 microgram concentration of the extract can carry out inhibitions of Staphylococcus, that is Typhimurium as well as Bacillus subtilis. Other pathogens require major concentration of the extract rather than these two concentrations. So this is the table which shows that these Pseudomonas aeruginosa and fungal pathogens like Candida albicans and Rhizomycor mehi can be inhibited by the methanol column fractioned extract compared with the column fractioned chloroform extract. So comparatively, the methanol column fraction was good enough to carry out the inhibitions of both the bacterial as well as the fungal pathogens. Therefore, this pathogen was, therefore that particular fraction was used to analyze, characterized with the used for characterization, as well as this particular compound were also compared with commercially available antibiotics, to what extent this particular compound can inhibit or correlate with the commercial antibiotics. So there too, the gonadal extract have the best activity against certain pathogens, as like that of gentamicin and penicillin. With the particular pathogens. Then this is the diagram which shows that the relationship of the commercial antibiotics with the crude gonadal extract, where you can see that the gonadal extract was good enough to compare with the commercial antibiotics. Therefore, the gonadal extract was again taken to characterize, that is the gonadal column fraction extract was taken for characterization, like analysis characterize a particular compound, where we can see that FT-IR spectrum of the sample give strong broad band at 3272.01. Hence, it can be concluded that the molecules has OH group as well as they have a strong NH group. The low carbonyl band and the NH band strongly suggest the presence of.
Ma'am, can you hear me?
Yes,
please, yes, ma'am, there's under 1 minute only.
Yes,
so
there is a presence of an amide group. Conclusion is that current study has investigated the potential bioactive effects of crude extract of the particular sea urchin, and it is good enough to carry out the sidal effect of both bacterial pathogens as well as the fungal pathogens. The crude extract was column fraction. The column fraction were also analyzed compared with the commercially available antibiotics, where these fractions were good enough to carry out the inhibitions of both the fungal as well as the bacterial pathogens as with the commercial antibiotics. Further on, further studies have to be carried out on this particular column fractioned sample to elucidate the structure of the most effective bioactive substance. Like we can go with the GCMS, MS, NMR, and other studies to exactly locate the particular compound. Thank you.
Thank you, Ms. Sanijan. I have a question that as we know, the target for bacteria, right? Either it acts on cell membrane or ribosome, and target for fungus differs. So how you confirm that your target is beneficial for fungus as well as bacteria both?
It can definitely interfere with the cell membrane mechanism, ma'am.
Cell membrane. So it is effective for the cell membrane of, you mean to say both bacteria and fungus?
Yes,
Because the cell membrane content of both differs.
Yes, ma'am. Yes, cell membrane differs for both fungi and bacteria. But actually, with the bacteria, normally cell membrane metabolism is highly inactivated or it may interfere with the mechanism. But when it comes to fungi, once the mechanism is being inhibited, the fungi cannot tolerate that particular condition.
So you need to add anything else to make it more efficient antifungal?
And yes, ma'am, more studies has to be concentrated on this. When it comes to fungi, we have to go
because
in depth
both
into
has different structure, different chemical composition, and one compound cannot be compared. So you have to add something else to make it better antifungal.
Yes, ma'am. As like I said, future studies have to be concentrated on such aspect that because we have eluted a particular compound, column fraction compound, but studies have to be elaborated to come to a conclusion that this is the exact mechanism behind inhibiting such a pathogens.
Okay. Okay. Thank you.
Thank you, ma'am.
Thank you, ma'am. Dr. Ram Babu Sharma, are you there, sir? Are you free for your presentation now?
Yeah. Good afternoon, ma'am. I'm here.
Good afternoon, sir. Please share your screen.
Yeah. I would like to thanks BioLEAGUE for giving me a platform. At first, good afternoon, ma'am, and good afternoon, other participants. Hello, I'm audible, ma'am?
Yeah, hi, good afternoon. Yeah, please go. Please
The topic of my studies a socio-economic epidemiological characterization of strep typhus positive cases by clinical and laboratory findings at a tertiary care teaching hospital, southern part of Rajasthan, India. So I start with introduction. Strep typhus is a zoonotic infectious disease caused by a bacteria or Escherichia species. Human can be exposed by through bite of infected larval stage of tuberculous bite, which are found in rodent of forest and rice field across the Asia Pacific region. In India, the first case of strep typhus was detected in Assam in 1930. Simultaneously, after Second World War, the positive cases was also reported from Himachal Pradesh, Tamil Nadu, Kerala, Maharashtra, Bihar, Karnataka, and Rajasthan. West Bengal and other two states, they are highly endemic for strep typhus cases. The trombiculid might have a metamorphic life cycle. The female might lay fertilized egg in soil, from which 5 to 7 days later, 6-legged larva hatch out. The proliferation of the bacterium occur at the site, forming a characteristic skin lesion known as the eschar. After incubation of 10 to 12 days, the affected person experience headache, fever, anorexia, and general lymphadenopathy. The additional symptoms in strep typhus infected patient, the enlargement of the spleen, nervous disturbance, delirium, and prostration. The mortality range of strep typhus is around 6 to 10%. Death occurs as a direct result of the disease or from secondary effects such as pneumonia, encephalitis, and circulatory failure. The absence of eschar, the strep typhus specific symptom, make clinical diagnosis more challenging. The disease severity and manifestations vary widely from asymptomatic to fatal and show marked geographical differences. So it is the main reason for new cases arrival in a certain geographical area. The general course of the disease and the prognosis vary considerably depending on the character of endemic strain. Therefore, early diagnosis is very essential to start treatment and prevent further complications. The need of the study, the purpose and need of the present study were to identify the challenges in epidemiological and clinical profile, clarify current diagnostic issues of strep typhus, arouse clinical awareness, and facilitate its diagnostic and preventive measures. The research question in my study is that what are the optimal testing strategy for diagnosis of the common causes of acute underdiagnosed febrile illness in southern part of Rajasthan, India. The main hypothesis of this study is that current diagnostic tests, usually based on the paired serology for common known malarial cases of fever, exclude scrub typhus infection in southern part of Rajasthan, are inadequate for clinical use and lead to underdiagnosis of this infection. The diagnostic accuracy and the clinical value of using rapid tests need to be determined to diagnose causes of fever in a rural clinical setting. The plan of this hypothesis will be answer the following questions. What are the causes of acute underdiagnosed febrile illness in patients presenting with fever where a definitive diagnosis cannot be given in the field, excluding malaria and dengue cases? How can we best diagnose these diseases in the field with a clinically useful turnaround time? And can new generation rapid test for scrub typhus infection be used to reliably determine the cause of infection in patients with acute undiagnosed febrile illness in a rural area? Can new molecular diagnostic assays replace current serological methods as gold standard test for confirmation of these infections? The main aim of the study is to evaluate the clinical picture of the symptoms observed in the scrub typhus positive cases in the given area of Rajasthan. The principal objective of my study is to determine optimal testing strategy for the scrub typhus infection to determine the diagnostic accuracy and clinical value of using new generation rapid tests, ELISA, and molecular diagnostic assay to diagnose causes of fever in the given region. The second objective is to improve the understanding of these infection in the population to assist the development of the treatment algorithm and patient management for underdiagnosed acute febrile illness cases. The study is conducted in the Department of Microbiology and General Medicine in PIMS, Udaipur, Rajasthan. The total duration of my study is 24 months and it is prospective and analytical study. The inclusion criteria of my study is the all age group patient with acute underdiagnosed febrile illness presenting with or without scar in our hospital will be included. And the exclusion criteria is all other patients with tropical illness such as malaria, dengue, typhoid fever, brucellosis will be excluded in the study. All duplicate patients will be excluded from the study. The material and methods the scrub typhus was confirmed by IgM ELISA according to manufacturer's instruction. And after confirmation of scrub typhus, a detailed clinical assessment of each scrub typhus positive patient for the sign and symptoms, including a careful search of an scar and basic other laboratory findings. They are documented by using a standardized form. The result and discussion that in the result, the gender distributed among the scrub typhus positive patients, the highest prevalence was found in female patients for 55.71% than in male, where is the 44.29%. That
Sorry
age
to
group
interrupt you, sir. 2
Yes.
more minutes, sir.
Yeah, ma'am. Age group distribution among these patients, the higher prevalence was seen in 41 to 50 years, then followed by 31 to 40 years. Total 6 patients was diagnosed with scrub typhus. The distribution of patient according to presenting complaints, fever was recorded 100% in patients, followed by headache 88.55% patients, nausea in 62%, arthralgia 53%. Thrombocytopenia 46, breathlessness 51%. SCAR was found at nearly 23.71% patients. The SCAR was seen in male patients 23.2% and in female 24.1%. The socio-demographic characters was distributed among these patients. 15% was urban population, whereas 84% was rural population. In other, based on occupation, 11.8% was housewife, 6% was drivers, 8% were students, and the majority of the cases are 68.57% are farmers. This is the main reason to found new cases of scrub typhus in this given area because majority of the patients work are agricultural. The scrub typhus is a, we know very well, it is a deadly infectious disease that affect about 1 million people each year. It is a seasonal disease with high incidence in the post-monsoon season. All diagnosed patients develop symptoms between July and November in my study. The 87% of these cases occurred from September to November. Similar findings have been reported in other studies like Kamasara The seasonal pattern should alert treating physician to consider strep typhus on list of the differences that typically only include dengue and malaria during these seasons. In this study, the majority of the patient was recorded 100% in fever. The similar study was done by Burgess et al. Found the most common symptom as fever, nausea, vomiting, 54%, shortness of breath, and headache, cough, and paresthesia. Vijay Kumar also stated that they conducted 150 patients had neurological symptoms, headache, neck pain, seizure, weakness, or altered sensation. And summary in conclusion, strep typhus is a serious acute febrile disease with significant mortality. Respiratory failure, shock, acute renal failure, and MODS are serious life-threatening complications of this disease. Strep typhus fever occur in common area with disease which have similar clinical manifestations like other malaria, dengue, fever, typhoid, and leptospirosis. However, rising awareness of these infections among clinicians in endemic area and developing reliable method for faster diagnosis. One of the limitation of my study is that strain variability and virulence of infection strain previously reported to be associated with strep typhus were not examined. Therefore, further studies focusing on the type of circulating strain are essential to obtain a complete clinical profile for this infection. The study is designed from a single medical center and possibly biased in patient selection or referral and incomplete epidemiological data available of the patients. Thank you, ma'am. Hello.
Yeah, thank you. Swati ma'am, are there any questions
Yes,
for?
yeah. Dr. Rambabu, nice presentation.
Yeah. Thank you, ma'am.
I just want to know what were the risk factor in your patients?
Ma'am, certain patient showing the other laboratory findings are their liver enzymes were also altered. SGOT and alkaline phosphatase was raised, bilirubin was also raised, and in lipid profile, triglyceride level was also raised. And they admitted in our hospital with complaints of severe breathlessness. And after I was not put up my one more finding that 22% patient mortality was recorded in my study because ये मैंने ये publish नहीं किया अभी present नहीं किया क्योंकि इससे अभी मुझे
Yeah, that was my next question.
Yes, ma'am.
What was the mortality in your patient with you, and how many was died because of this infection? So you answered 22%.
Yes,
Okay.
ma'am.
And
I was
your
saying
demographic data is very wide. You have a housewife, you have agriculturist. So I just wanted to know how these people are. I can understand the agriculturist or forest worker are exposed. How these housewife or working inside home are exposed to scrub typhus infection.
Ma'am, 3% housewives are infected with scrub typhus. 3%, ma'am.
What was, how did you find out the history? Like how
Yes,
they
ma'am.
got,
Ma'am, some female patient was migrant.
Okay.
Like she was working another region, then she was come into our hospital.
So they were
with
not strictly housewife. They have exposure to outside, right?
Yeah, ma'am.
Okay.
Yeah, ma'am. Because it
Okay.
is only a single hospital based study, ma'am. If I go for a large scale, then we can
No,
find
that I think
more valuable data.
450 sample size is enough to give evidence. I don't think so. It's good to have larger, but 450 is good enough to give conclusive data. Because I'm not sure how much is in your setup, in your place which you stay. There is more cases of scrub typhus reported. Because where I stay, there are rare cases of scrub typhus reported.
Ma'am, my area where I'm working here in a medical college, this area is the most harbor area of scrub vegetation because it is surrounded by a hilly region, and every person working as in the agricultural work. So only 3 to 4 studies done previously 6 to 8 years in Rajasthan, ma'am. And this my present study is I want to study more their strain variability. I will go for strain variability, ma'am. which strain are more prevalent in our area? Because a patient is positive with scrub typhus in second time. If previously he was positive or she was positive with scrub typhus, then which strain was infected? And new after 1-2 years, he or she was again infected with scrub typhus, then which strain are responsible? So it is the main reason, ma'am, to various other complications were found in my study.
Okay, thank you. Thank you, Mr. Sharma. Thank you.
Sir. For the next presentation, I invite Ms. Chandana Chindu.
Yes. Am I audible?
Yes, very audible.
Okay. I'm sharing my screen. Okay, it is visible, right?
Yeah, it's visible.
Yeah, okay. Good afternoon, everyone. My name is Chandana Chintu, research scholar working under the guidance of Dr. Kiranmai Patnala, Associate Professor, Department of Biotechnology from Geetham University, Visakhapatnam. So the title of my research topic is Mycoremediation of Crude Oil by employing native fungi isolated from the most common oil contaminated sites along the Visakhapatnam port. Okay. So crude oil is an indispensable chemical raw material that is mainly used as a fuel, and the majority of the crude oils produced all over the world are mostly transported through coastal and marine environments, making the leakage of oils from the containers carrying these crude oils as a major source of contamination in marine environments. So oil spillages are considered to be the accidental or unintentional release of liquid petroleum hydrocarbons into the environment, and it is estimated that about 706 million gallons of oil enters the oceans every year, with about 32% contributing during the loading and discharging of oils, and for about 28% contributing to the spillages that usually occur during the collision of these oil tankers. So whenever there is a collision or a oil spillage, there are various physical, chemical, and mechanical methods that are usually employed. These include in situ burning or mechanical recovery, usage of skimmers, sorbents, and dispersants. However, these conventional methods are said to be having their own disadvantages, like being they are very expensive and they are labor intensive, and with the main concern is that they mainly disturb the marine ecosystems. So a more sustainable, eco-friendly, and cost-effective process needs to be employed where bioremediation comes into action. So bioremediation is the use of microorganisms for clearing these contaminants or breaking down these contaminants. So for our research study, we have been using marine fungi. Why we have opted for marine fungi is there salient features of their enhanced ability to tolerate crude oil and sustain even in these contaminated sites. The presence of their elongated hyphae make them to spread for larger areas and provide more surface area for biodegradation, and also their ability to tolerate and adapt to the fluctuations of pH and temperature. And Visakhapatnam, as we all know, is one of the major ports that is involved in loading and discharging of oils, which is known to be contributing for about 32% of oil contamination in marine waters. We have mainly combined these two factors of employing marine fungi to degrade these spillages that usually occur in and around the Visakhapatnam port. So the main aim of this study is to identify potential fungal strains that can degrade these hydrocarbons without usually disturbing the marine ecosystems. So keeping this aim in mind, we have designed 3 objectives. With the first one is to isolate and purify the strains that are already able to grow and sustain in these polluted sites. Later on, to assess their capability to degrade, assess their capability to utilize these oils as a source of carbon, and finally to determine their degradation efficiency, like to what efficiency they are able to degrade these petroleum petroleum hydrocarbons. So by keeping these objectives, we have formulated a methodology where we'll be initially collecting the samples, marine waters from the oil contaminated sites. We'll be culturing them. We'll be taking out the marine fungi and performing the degradation assays. So the methods that are employed are spread plate and streak plate for isolating pure cultures, polyphasic for identifying the fungal strains that are able to degrade these petroleum hydrocarbons, and performing DCPIP degradation assay method to detect which are able to degrade these oils. So for our first objective, we have employed 3 collection sites, oil refinery berth 1, 2 and naval dockyard. So along the Visakhapatnam port, we could see major oil contamination in these 3 sites. So we have collected the water sample from these sites. We have transported the laboratory at 4 degrees centigrade and performed the experiment. So we have taken 4 different type of media: sabrodextrose agar, potato dextrose, malt extract and Zepax agar media. And in order to get different strains, different variants of fungal strains. So as a result, initially we have obtained various varied varieties of fungal strains, of which a total of 65 pure fungal strains we were able to successfully isolate from these sites. From these 3 sites, we were at the last able to successfully isolate around 65 pure fungal strains. For these 65 fungal strains, up to now we have assessed their potential to degrade crude oils for about 47 strains. So among those 47 strains, 39 strains have shown very minimal or no degradation, 51% have shown a medium degradation, and the 10% have shown a major degradation ability. So we have concentrated on these 10% of microbes, which are accounting for about 5 microbes. Among these 5 microbes, one particular strain was known to exhibit a very high potential of 82.13%. The other 4 strains have exhibited a potential ability to degrade of about 69% to 76%. So for this degradation assay, we have mainly gone through DCPIP assay, where the redox dye of DCPIP will change from blue to colorless when there is a degradation of oils by the microbes. So keeping that in view, we have performed the decolorization percentage, and along with that, we have also weighed the fungal dry mass, the growth,
Can you
whatever
move
is.
through a little faster? Can you?
Yes, ma'am. This is almost done. We have weighed the microbes, and both these data have correlated, and it has shown that one of the strains has been exhibiting a very high potential. So the research is still in its initial stages, and we are looking for any other microbe or fungal strain which can degrade to a higher potential. So as we got some promising results with these findings, we shall be further continuing our research and look for better degrading strains. So in summary, the research aimed to assess the effectiveness of fungal strains in mitigating oil pollution, and it investigated the potential of marine fungi to degrade petroleum hydrocarbons. So I extend my heartfelt thanks to my guide, Dr. Kiranmai Patnala and Geetham University, for funding this project. These are my references. Thank you. Hello.
Nice presentation, Ms. Chandana.
Thank you, ma'am.
Ms. Chandana, have you tested those against those your fungal strain, and
yes,
you have tested and verified?
Yes, ma'am.
Okay, so what was, can you repeat what was your interpretation?
Like we are looking for the microbes that can degrade these petroleum hydrocarbons or these crude oils, ma'am.
So you were able to, you were successful what you were working on?
Yes, ma'am. For 82%, one of the strain has exhibited highest potential, contributing to about 82%.
Okay, so you have any plan further to take it up?
Yes, ma'am. Because for the 65 fungal strains, I have performed analysis as of now for 47. We are looking for somewhat more promising fungi strain, ma'am. If we get a more promising strain, we are planning to look deeper into its metabolic pathways, or how we can actually implement in the open marine environments.
Okay, thank you. Thank you, Ms.
Thank you, ma'am.
Thank you, Ms. Chandana.
Thank you.
For the next presentation, I invite Ms. Neha Rawat for the poster presentation.
Good afternoon, ma'am. Is my voice audible?
Yes, ma'am.
Shall I share the screen?
Yes, please.
Just wait a second. Is my screen visible to you?
Not yet.
Now?
Not yet. It says you have started screen sharing, but we are not able to see the slides yet. Yeah, we can see the slides now.
Okay. So good afternoon, everyone. My poster presentation is on the topic biofilm production by Pseudomonas aeruginosa and its association with multi-drug resistance in clinical isolate. Basically, it is a review based on several studies, and it is a part of an undergoing research project. So, first of all, to begin with the introduction, biofilms are the aggregation of bacteria which are enclosed in extrapolymeric matrix. It shields the bacteria from any type of resistance such as antibiotic, disinfectants, and from host immune response. So it basically acts as a survival mechanism for the bacterial communities. And also biofilm is attached to biotic or abiotic surfaces, which contributes to the pathogenesis of the infection. According to National Institute of Health survey, it showed that in 65% of microbial infections and 80% of chronic infections, they have been commonly associated with the development of biofilm. Pseudomonas aeruginosa is an excellent model to study the biofilm as it is an opportunistic pathogen. Because of its ubiquitous nature, it causes nosocomial infections and it has potential to form stubborn biofilm in indwelling medical devices. So many virulent factors in Pseudomonas aeruginosa which contributes to the biofilm development such as pili, flagella, edicin, and exopolysaccharides. So the current review, it aims to understand the association of biofilm with multi-drug resistant strain of Pseudomonas aeruginosa. Why the study is needed? Because of high mortality and morbidity rate seen in Pseudomonas aeruginosa infection, it decreases the patient's quality of life and it increases the economical burden. The multi-drug resistant strain, they promote the biofilm formation as seen in patients with chronic stage of infection such as in ICU and patients with implanted medical devices. Third, the thing is there have been antibiotic therapeutic failure because biofilms are resistant to common anti-pseudomonal drugs. So the next is materials and methods. Several research journals and web-based sources were reviewed, and it included only the peer-reviewed articles which were published between 2017 and 2023. Okay, the searching databases were PubMed, Scopus, Web of Science, ScienceDirect, and the keywords used were biofilm, Pseudomonas aeruginosa, multi-drug resistant, quorum sensing, and antibiotic susceptibility. For the method, a graph was plotted to demonstrate the association of biofilm development with multiple drug resistance. And all the researches which were included, they assessed microtiter plate assay for biofilm detection and Kirby-Borders diffusion method for antibiotic susceptibility pattern. So for the result, a graph was plotted for the association of biofilm with MDR in different studies from 2017 to 2023. The graph demonstrated that majority of Pseudomonas aeruginosa isolates, they form biofilm, and the maximum isolate forming the stronger intermediate biofilm, they were found to be multi-drug resistant. That is, they were resistant to more than 2 classes of antibiotics. Very few studies demonstrated that isolates with weak or none biofilm producing capacity, they were multi-drug resistant. So it can be observed from the above studies that MDR is associated with strong biofilm production, and the mutation in the bacterial genome as well as the exposure of bacteria to high levels of antibiotics, such as inappropriate irrational use of antibacterial drugs, it causes multiple drug resistance in biofilm. So hence the summary, Pseudomonas aeruginosa, it leads to prolonged am I audible?
Yeah, you're audible, and there's about half a minute left for presentation.
Okay.
Yeah.
So basically, in conclusion, the patient with implants and surgeries are more prone to the infection caused by Pseudomonas aeruginosa, as it is observed among all the clinical isolates, including IMDs. Large number of studies should be reviewed, and genomics and in vivo methods should be employed in researches. New antibiofilm strategies are necessary to handle biofilm-associated chronic infections, as antibiotic therapy is not effective once biofilm is matured. So next are the references. It is very short. It was very short subtopic. So thank you for giving me the platform to present the topic. Thank you.
Thank you.
Thank you, Ms. Neha. Yeah, I have a few questions. What is biofilm?
Ma'am, biofilm, actually what happens, bacteria, they collect with each other. They start communicating with each other by signal production, and they secrete matrix, extra polysaccharides. So what happens? Exopolysaccharides. So what happens? Debris is collected and extracellular DNA is collected, and they form a biofilm.
So who all are risk patients for biofilm production in a hospital?
Which are more at risk?
Yeah, which all patients are more at risk?
The immunocompromised population, or the patients with surgeries, or for example, burn and wound patients, or catheter associated.
Yes, the patient who has external devices, plastic devices,
Yes,
right?
ma'am.
That is the
Yes,
surface
ma'am.
which, yes.
Yes,
Which
ma'am.
other bacteria apart from Pseudomonas is for biofilm?
Ma'am,
For biofilm
staff,
production.
apart from Pseudomonas aeruginosa, the biofilm is can be produced by Staphylococcus aureus and Klebsiella pneumoniae. So,
any fungus?
Right. Yes, ma'am, there are fungus.
Candida?
Candida albicans.
Okay, thank you, Neha. Thank you, Ms. Neha.
Thank you so much, ma'am. Thank you.
Thank you, Ms. Neha. For the next presentation, I invite Ms. Hemlata.
Yes, ma'am.
Hi, you're up. Are you ready with your presentation?
Yes, ma'am. Yes, I'm
please
ready.
go ahead.
Am I audible, ma'am?
Yes, ma'am.
Okay, ma'am. Good afternoon, all. Good afternoon, Swati, ma'am. I am myself Dr. Hemlata from Guntur Medical College, Guntur. Today, my topic is zero prevalence of hepatitis A, B, C viruses in a tertiary care hospital, Guntur. My co-author is Dr. K. Parmeshwari, ma'am, who is a professor and HOD of Department Microbiology. It's my poster presentation. Is it visible, ma'am? Is it visible, ma'am?
Yes, yes.
Okay, ma'am. Coming to the introduction, the term viral hepatitis refers to a primary infection of the liver by any one of the heterogeneous group of hepatitis viruses, namely hepatitis A, B, C, D, and E viruses. And these viruses are taxonomically unrelated except for type B, which is a DNA virus. All others are RNA viruses. The features common to them are their hepatotropism and ability to cause a similar icteric illness, ranging in severity from unapparent to fulminant form. Viral hepatitis overview is a overview. The source of infection for A and E are feces, and for B, C, and D are blood and blood-derived body fluids. And route of infection for A and E are feco-oral, and for B, C, D are percutaneous and permucosal route. And B and C, D causes chronic infection, and whereas A and E will not proceed to chronic infection. The prevention for A, B, and D is pre and post exposure prophylaxis, and whereas for C, we can do it for blood screening and risk behavior modification, and for E, it is ensure safe water drinking. These are the structures of hepatitis A, B, D viruses, and hepatitis C virus. And the risk of chronic infection of hepatitis B virus is determined primarily by the age of acquisition of infection, being much higher when the infection occurs in infancy or early childhood, and below 5% when it occurs in adults. Hepatitis D virus can cause infection only in the presence of hepatitis B. Hence, an individual protected against HBV is also protected against HDV. Hepatitis E can cause fulminant hepatitis rarely in 1 to 2% of the cases, except for the pregnant women who are particularly at higher risk, that is 20% for developing fulminant hepatitis. My objective is to study the seroprevalence of hepatitis A, B, C, and E viruses in tertiary healthcare hospital. My materials and methods include it's an observational retrospective study performed from January 2023 to July 2023 at our tertiary care hospital, Guntur. A total of 333 samples were collected and were tested for hepatitis A, B, C, and E using enzyme-linked immunosorbent assay kits, that is diapro. My results include, out of 333 samples, 93 were tested for HAV, in which 19 were found positive, 20.43%, and out of 19 positive samples, 10 were children, accounting for 52.63%, and 5 were males and 4 were females, 26.31% and 21.05% respectively. Out of 333 samples, 93 samples were tested for HEV, in which the positivity is 0. Out of 333 samples, 109 samples were tested for HBV, in which 35 were found positive, 32.11%. Out of 35 positive samples, 22 were males and 13 were females, 62.85% and 37.15% respectively. Out of 333 samples, 38 samples were tested for HCV, in which 11 were found positive, and out of that is 28.94%. Out of 11 positive samples, 72.8 were males, 72.72%, and 3 were females, 27.23%. And the pie chart showing HBV is 54% and HCV accounting for 70%, HEV is 0%, HAV is 29%. Coming to the gender prevalence, HAV more male preponderant, and HAV is
Sorry,
more
Ms. Hemlata, the time is running out. You have to wrap it under 1 minute. Am I audible?
Okay.
Yeah,
HAV is mostly seen in 10 children at 52.63%. HBV is more in males, 22%, 62.85%. HCV is more male preponderant, 8, and HEV is not seen anywhere. It's a bar diagram showing HAV mostly in children, more seen in children. HBV in male preponderant, and HCV is also male preponderant. Coming to the conclusion, the prevalence of hepatitis B was high before the implementation of a universal vaccination program, and it has been decreased later years. And the prevalence was higher in rural areas than urban areas. One study showed that HAV and HEV were the major cause of acute hepatic failure due to the fecal contamination of drinking water. Hepatitis E virus infection positivity is nil at a good note. Zero prevalence of HCV in our study is 17%. WHO estimated that in 2019 approximately 290,000 people were derived from hepatitis C, which is a cause for cirrhosis and hepatocellular carcinoma. Unfortunately, there is no effective vaccine for hepatitis C. Our GGH is running serological tests for hepatitis C under guidelines of National Viral Hepatitis Control Program. Enormous vaccination awareness programs or communication strategies are necessary, still needed to reduce the highest positivity rate, ma'am. These are my references, ma'am. And thank you, ma'am. Thank you, ma'am.
Thank you, Dr. Hemlata.
Thank you, ma'am.
So you were saying in your result, patients are positive for hepatitis B or A, right? I just want to know which technique they were positive. What technique did you use to say that these many percentages are positive? They were PCR positive?
No, madam. ELISA, madam. Only ELISA, madam.
That you need to mention. That
Okay,
need to
madam.
mention. None of the
Okay,
result you have said which technique and which
Okay.
ELISA you have used, which type of direct, indirect.
Okay, madam. That's a
Yes. Yeah. So please,
Okay.
that all need to be included, and in results also you need to mention ELISA and which IgM or IgG. What did you detect, or you detected both?
IgM, madam. Only IgM, madam.
Only IgM.
IgM, madam.
Okay. Okay. Thank you. Thank you, Dr. Hemlata.
Thank you. Thank you, madam. Thank you.
Thank you, Ms. Hemlata. For the next presentation, I invite Dr. Shrutika Goel.
Hello, ma'am. Am I audible?
Yes, I can hear you.
Okay. Ma'am, is my screen visible?
Not yet, ma'am. Yes, ma'am. It's visible now.
It's visible. Okay, one sec. Ma'am, is it visible?
Yes, ma'am, it's visible.
Good afternoon, all. So the topic of my presentation is dentistry after COVID era, which is a review. So we all know that COVID-19 had struck the world in all aspects, but here we'll talk about dentistry. COVID had said to be a highly communicable disease, and the route of transmission was air or water droplets. In dentistry, dentists and other dental healthcare professionals usually comes in close contact of the patients in day-to-day life. So the treatment offered in dental care procedures produces aerosols that are proven to contaminate or as source of transmission of many diseases, and so as for COVID. So therefore, many preventive measures were brought into being, and many of them are still followed because it has somewhere changed the aspect of dentistry. So here we'll discuss about a few of them. The first that we do that has changed dentistry is the assessment of form for medical history and evaluation for signs and symptoms of COVID in patient post-COVID signs and symptoms. The protocols to be followed by the dental professionals are airborne precautionary measures like PPE kits, hand hygiene practice, head cover, shoe cover, N95 mask, face shields, or eye protective glares. Indoor portable air cleaning system should be introduced. Pre-procedural mouth rinses by povidone or hydrogen peroxide in a concentration of
I think there was a technical issue.
Yeah, we'll just give a minute for Dr. Shrutika to rejoin.
Sure, ma'am.
Ma'am, is my screen visible now?
Your screen is not visible, ma'am.
Ma'am, can you just give me 1 sec? Ma'am, is it now visible?
No, ma'am. Earlier, I think you were logged in 2 devices. Now only one is shown now.
Actually, ma'am, my laptop is.
Okay. Do you want to fix the issue and come back? We can do the final presenter and then we can give you the chance. There's just one more.
Okay.
In the meantime, you can figure out how to share screen. Yeah. Okay. Dr. Jyoti Vyas.
Hello, I'm audible, ma'am.
Yes, ma'am, you are audible now. Yeah.
Ma'am, my screen is visible
Yes,
now.
ma'am, your screen is visible. Into the picture.
Good afternoon, ma'am. Myself, Dr. Jyoti, Junior Resident, Department of Microbiology, UPMS, FA, Etaba. My topic for poster presentation is prevalence of candidemia in a tertiary care hospital, Western UP.
Ma'am, can you do slideshow? Your screen is, we cannot see fully. Or you can zoom. Yes. But when you zoom, the letters are hazy. Try one time slideshow. I cannot read.
Ma'am, it is visible now. Hello. Ma'am, it is visible. My slide is
down.
visible.
It is there slideshow, right side down. Right side down. In your screen.
Ma'am, please magnify, turn on the zoom option, ma'am. The time is also running out.
Okay, please go ahead. Go ahead. Dr. Jyoti?
Ji, ma'am. Introduction. Candida species are ubiquitous yeast but opportunistically invasive in individual whose cell-mediated immunity is decreased due to any disease condition or by any iatrogenic intervention, mostly observed in ICU, oncology department, and organ transplant department and dialysis unit. Candida bloodstream infection have become a major problem in tertiary care hospital worldwide. Candidemia has been observed particularly among patients hospitalized for long period who have been exposed to antibiotics, immunosuppressive therapy, parenteral nutrition, and multiple invasive medical procedure. Need for the study, there is an increased prevalence of candidemia worldwide, including India, which is a major cause of concern for clinicians, especially in severe underlying causes. The aim of this study to determine the prevalence of candidemia in tertiary care hospital in UP. This is the data collected from our institute in UPMS FI Etava in patient of candidemia from March to August in following percentage in 2023. Methods and material. This retrospective study is conducted in all age group patients from all indoor patient department of tertiary care hospital in Western UP. The study was conducted between March 2023 to August 2023 in UPMS FI Etava. The conventional phenotypic method were used to identify the isolate of Candida species obtained from blood culture. An isolate of Candida obtained from blood culture and following methods such as microscopy, germ tube tests were used. All statistical data analysis using SPSS version 22 software. Result and discussion. Out of 2,330 blood samples, 38 isolates of Candida species were obtained in all age group patients from all indoor patient department in UPMS Safai, Etawah. Over the past 30 years, numerous investigators have reported that the frequency of severe infection caused by yeast, especially Candida species, has increased dramatically. Emerging non-albicans Candida species showed increased virulence, increased mortality, and resistance to controlled common antifungal drug. Hence, species identification is very important. Candida albicans has been the predominant isolate species. This increased prominence of non-albicans species causing bloodstream infection have been extensively noted in other Indian studies also. The high incidence of Candida bloodstream infection among patients at the extreme of age, neonates, and age group more than 45 year accounted for highest number of isolates. Many risk factor and comorbid conditions have been reported in the development of candidemia in our study. More severely ill patients are at higher risk for Candida infection. Candida bloodstream infection is an important cause of mortality and morbidity. Summary and conclusion. Candidemia remain a major cause of morbidity and mortality in the healthcare setting in this study. Candida albicans is the most common cause of candidemia, whereas the non-albicans Candida species are the major cause of candidemia in neonate group. Early implementation of medical intervention to reduce the incidence of candidemia in all age group. Candidemia extend the duration of hospital stay and increase the cost of medical care. These are the references.
Thank you, Dr. Jyoti. So your duration of studies were
6 months.
6 months. So in 6 months,
March to August 2023.
in 6 months, you were able to collect how many samples?
Samples were 2,330 blood samples.
So they were ruled out for bacterial septicemia. Then you went ahead with fungal septicemia, or how it was?
Ma'am, directly fungal septicemia, candidemia.
So they were having any a bacterial plus fungal infection or only fungal infection?
Only for fungal.
And you said non-albican candida you saw in neonate, right? Yes. Which non-albican candida?
Ma'am, Candida glabrata, and Candida tropicalis.
How you speciated those candida?
Ma'am, on chromagar by different color.
But so many times the color will be misleading, right? So did you go ahead with any molecular technique?
Molecular technique, ma'am.
PCR or anything else?
Ma'am, by PCR.
You did PCR for Candida for those positive samples, or those 38 samples?
Ji, ma'am. Conventional PCR.
Conventional PCR. Okay. Any mortality in your case, in your studies?
Mortality was, ma'am, 10 to 20%, ma'am.
10 to 20 is very wide range.
Ma'am, actually, it was not
sure. So you just check what was the mortality. Follow up your patients and just check whether patient improved or deteriorated.
Okay, ma'am.
Thank you, Dr. Jyoti.
Thank you.
Thank you, ma'am. Thank you. Dr. Shrutika Goel, is the issue sorted?
Ma'am, yes, I'm trying to share the screen again.
Okay. Hello.
Ma'am, just give me one second. Can you see my screen?
Yes, we can see your screen, ma'am. Yeah, we can see.
Is it visible now?
Yes.
Okay. So, ma'am, as I was saying that there are many protocols that have been taken in dentistry after the during and in fact it is followed now also after the COVID scenario. So a few of them are wearing PPE kits, having a proper hand hygiene, shoe cover, N95 mask, face shields. Then we have installed indoor portable air cleaning system. Then we advise patients for pre-procedural mouth rinses with povidone 0.2% or hydrogen peroxide rinse 0.5 to 1%. Then we use disposable devices as much as possible. There is a proper rubber dam isolation for any kind of treatment as much as possible. We minimize the use of ultrasonics or handpieces, rotary handpieces, which is the main source of production of aerosols, which can again increase the chances of contamination from the patient to the dentist or from the dentist to the patient. Then use of anti-retraction handpiece, and wherever possible, we use hand instruments. We use hydrochloroquine for prophylaxis, disinfection of the settings with sodium hydrogen hypochlorite, and proper management of the medical waste, and the installation of negative pressure treatment room. Thank you.
So, Dr. Shruti, you still follow which all measures you mentioned just now?
Ma'am, the use of PPE kits and face shields and N95 masks are the most prevalent that are followed. Ma'am, indoor we have a cleaning system at the clinics, because now there are many portable cleaning systems available, marketed, commercially they are available. And ma'am, what we advise our patients is apna pre-procedural mouth rinses we do advise patients. So it decreases the production of aerosols that usually occur when we do any kind of treatment.
Okay. Thank you, Dr. Shruti.
Okay. Thank you so much.
Thank you so much, Dr. Shruti.
Yes.
Thank you so much. And with that, we have come to the end of the technical session for the second international conference on clinical and applied microbiology. My sincere thanks to our session chair, Dr. Swati Kumari. Thank you so much, ma'am, for taking the time out of your busy schedule. And I know the ran a little late, but you've been so patient. You've given great feedback to all the participants in a very, very kind and polite way. Thank you so much, ma'am. I'm sure they found it very useful for their research. Thank you again so much, ma'am.
Thank you. Thank you, all of you. I hope the session was useful for all of you.
Thank you, participants. Definitely I have to thank you all for your involvement and your participation. Thank you all. Announcement about your certificates. For the listener category as well as the presenters, the certificate will be provided by the mid of next week, or like Wednesday or Thursday of next week, along with your conference proceedings. So if you have any queries in between, you can definitely reach me. Thank you all for the wonderful conference. Have a great rest of the day. Hope to see you also. Thank you.